huge round of applause, ladies and gentlemen. Thank you. I would again request Sri D.D. Jadeja, sir, to present a bouquet of flowers and a memento to Dr. Purnima Sharma. Welcome, ma'am. Thank you. Thank you, ma'am. I would now request Dr. Ansuya Bhadalkar to present a bouquet of flowers and a memento to Sri D.D. Jadeja. Can we have a huge round of applause, ladies and gentlemen? It is now my privilege to invite Sri D.D. Jadeja, IAS Mission Director, GSBTM, to deliver the welcome address and formally set the context for today's conclave. Sri Digvijay C. Jadeja, IAS, currently serves as Mission Director, GSBTM, Government of Gujarat, and has previously held various administrative roles within the state government, bringing valuable governance experience to the biotechnology sector. Sir, we request you to kindly address the audience. Can we have a huge round of applause for Sir, ladies and gentlemen?
Good morning, Namaskar. Warm welcome to you all. I have nothing more to say because you all are good researchers, clinical innovators, industry leaders and officials from BCL. So you all are authority in this field. But I welcome you all. Today's subject is cancer, and you all know cancer is leading cause of death globally. The primary purpose of this conclave is to advance cancer research and innovation for healthier lives, because we are funding agency for research, and society need good research. As today we are going to have one important collaboration with BCL, and we are going to sign agreement with them. I welcome Madam also on behalf of GSBTM. It is important to collaborate with private companies for innovation that promotes faster diagnosis and treatment of cancer. One young entrepreneur, just Ansuyaji introduced with me. So we welcome you all for this conclave, and I am hopeful good suggestions comes from you, and we are again welcoming you to this conclave. Thank you.
Thank you so much, sir, for setting the tone of this conclave. With a great honor, I now humbly invite Dr. Ashok Sharma, Additional Professor, AIIMS, New Delhi, to address the audience on the topic, Cancer Can Be Cured, Evidence from Modern Oncology. Dr. Ashok Sharma, PhD, is an Additional Professor in the Department of Biochemistry at AIIMS, New Delhi, and DBT Ramalingaswamy Fellow, as well as DST Ramanujan Fellowship awardee. A cancer biologist by training with a PhD in cancer biology from AIIMS, his work focuses on cancer epigenetics and translational cancer research, including biomarker development and gynecological cancers at the National Cancer Institute, NCI, India, AIIMS, Jharkhand campus. Sir, we request you to kindly share your valuable insights. Come on, everybody, can we have a big round of applause for him?
Sabhi ko namaskar. All the dignitary on the dais, and thank you for organizers inviting me for this wonderful initiative taken by the government of Gujarat. I'm heartily thankful, and this kind of event is definitely required to make awareness, because cancer, if we need to win the cancer, we need awareness. So, how the Joes? Lag nahi raha. How the Joes? Good. Honorable Prime Minister has seen the dream by 2047 we make a Viksit Bharat. But if our Joes is high, we make a commitment ourselves, we make 10 year back 2037. That will be able to achieve, but we need a high Joes and focus on the target. So as I discuss about the cancer curable, my slide is there. Okay, please. So we definitely fight against cancer and we defeated the cancer. You know, before my birth, even all of your birth, what are the main diseases in our country? कौन सी बीमारियां थी आज से 20-25 साल पहले, 30 साल पहले? कोई बताएगा? मेरा टॉक मोर इंटरेक्टिव टॉक ही रहेगा. आई एम नॉट गोइंग टू प्रेजेंट मोर लेबोरेटरी डेटा. तो कौन सी बीमारियां आपको लगता है 30-40 साल पहले थी? डायरिया, चिकन पॉक्स, कॉलरा, है ना, सारा इन्फेक्शन रिलेटेड. कोई आज उससे डेथ होती है किसी की? क्योंकि वी हैव अचीव इन द मॉडर्न साइंस. Scientific community working day night in the laboratory and strengthened our clinicians by technology wise, by medicines. So we defeated those diseases. But at present, in front of us is the cancer. There are two more diseases like cancer and neurological disorders. These are two main diseases in front of, because both diseases are depend on the longevity of life. Okay, so we need to address these two problems, and cancer is the main. Slide start हुआ? अच्छा, आप लोगों से एक क्वेश्चन मैं पूछता हूँ अभी, जब तक मेरी स्लाइड लगती है. आप लोग ये बताइए, वन डे वी डाई, ये तो फैक्ट है. कोई भी मतलब पूरा वो तो लेके नहीं आया कि मरेंगे नहीं हम लोग. तो 3 मेन रीजन्स जिससे हमारी डेथ होने का मैक्सिमम कारण हो सकता है. 3 ऐसे कारण जिससे हमारी डेथ होने के चांसेस बहुत ज्यादा है. कौन बताएगा? एनीवन? कोई बात नहीं, गलत होगा मैं ठीक कर दूंगा. कार्डियक अरेस्ट, वेरी गुड. सेकंड है एक्सीडेंट. थर्ड कैंसर. 1 2 3 में करें तो ये आपने सही बताया था, थैंक यू. देखिए 1 2 3 में फर्स्ट कौन सा? कार्डियक फर्स्ट, सेकंड कैंसर, थर्ड एक्सीडेंट. यहां थोड़ा सा मैं आपको करेक्ट करना चाहूंगा विद योर परमिशन. स्टडी पब्लिश इन लैंसेट 2019. This is the McMaster University carried out 21 low-income country, middle-income country study, epidemiological study. They found that cancer supersedes the cardiovascular disease. So cancer is number 1, because you see in every district hospitals and private hospitals stent easily available. मैं इंडिया के डेटा पे भी सर आपको फैक्ट प्रोवाइड करूंगा. कोई बात नहीं. So we are talking about the cancer like that. So some slide I will discuss. You see, these are the different diseases. Before my birth, or mostly people are sitting here. So we are supersede. We are already fight against that by multiple antibiotics. This is a paper which I am discussing about that. So this data from the low income country and middle income country. India has also come in that. Okay. So super like cardiovascular disease already supersede, cancer already supersede the cardiovascular disease, which I am saying. Sir saying ki this is a data of the globally, but India is that. Little bit I am agree with sir. Reason behind that, yes, incidence of cancer in our country is less, but population size is too high, so number of patients is very high, and infrastructure is less as compared to number of patients. If you see, that is the reason cancer is killing not just patient Indians, it also kills the India. Because one patient have the cancer, the whole economy you see is disturbed, whole family is disturbed. So it economically it also kills the India, our country. And these are the biggest challenge in front of the like high population size. You believe every year we add one Australia. इतना population हमारा हर साल increase होता है. Infrastructure उस ratio में we are not able to increase. And second thing, our economy now growing that public-private partnership is very less. We need to increase the public-private partnership to increase the fund. We strengthen our scientific backbone, especially in the cancer and research. Because you generally what we thinking when we make a policy, sir, this is for your request for you. When making a policy, we mop out the flow. We not try to stop the tap. If we want to stop the tap, we need to fund more R&D. And our PhD students, generally which after PhD what happens? They go abroad. We prepare one person and when he trained, he go outside the country. We should retain them in the country. How do we retain? We need to make a public-private partnership. Government already working very wonderful by startups. We need to more motivate the youngster to make a startup, small product which are importing from the outside. We need to develop here, and in that way we strengthen our clinicians by technology-wise, so we will give the better treatment for the patient. Now we have the resources, like less resources but population size very high. This is the data, the ICMR data. Highest incidence of cancer in our country is the Northeast. And lowest incidence is Maharashtra Barsi, 50 per million and 250 per million. In the same country, there is a huge difference. So this is a laboratory here. We need to understand those causes. What are the factors which are responsible to increase the high incidence of cancer in Northeast, but lowest in the Maharashtra Barsi? Okay, you see the slide here. This is the cause. This is a picture of the one house of the Northeast people. A 4x4 room, char-coated meat. So basically food habit, lifestyle, these are the major factors play crucial role to these like cancer and neurological disorder also. And you see, okay, one more question. Cancer is a good disease or bad disease? आप लोग क्या सोचते हैं? कैंसर अच्छी बीमारी है, बुरी बीमारी है? बताइए. अच्छी बीमारी है, कैसे? ये सर ने बिल्कुल सही बोला. हम लोग कैंसर से ना ऐसे डरते हैं, गली में से जब गुजर रहे होते हैं ना, एक कुत्ता पीछे लग जाता है. अगर आप भागोगे तो कुत्ता पीछे काट लेगा, तो वहां भागने की जरूरत नहीं है. अगर कैंसर किसी को होता है तो रुकने की जरूरत है, सोचने की जरूरत है, टाइम से अपना इलाज कराने की जरूरत है. एक ये जो डर है ना लोगों के बीच में, समाज से वो निकालने का बहुत जरूरत है. पीपल शुड बी अवेयर. कैंसर का दो इलाज, अर्ली डिटेक्शन, जितना जल्दी डिटेक्ट हो जाए, और जितना जल्दी उसका मैनेजमेंट स्टार्ट हो जाए. इफ वी फोकस इन दोस थिंग्स, तो सी 60% कैंसर आर प्रिवेंटेबल, 40% टोबैको रिलेटेड, 20% virus related. There are a lot of girls are here. How many girls have taken the cervical cancer vaccine? Raise the hand. कितने लोगों ने cervical cancer vaccine लगवाया? 2. देखो इतने मेरा 30-40 गर्ल्स हैं, लेडीज हैं, लेकिन 2 ने ही वैक्सीन लगवाया. ये हमारा इस लेवल पे आके जो ऑलरेडी हाईली क्वालिफाइड लोग हैं हम लोग, हमारे पास अवेयरनेस कितना कम है. So we need to spread this message also कि we should provide the vaccine, cervical cancer vaccine, 3 dose like COVID vaccine, so we prevent the HPV based infection. Second thing, like 80-20 formula, I thanks Dr. Professor D.K. Rath, he invented that formula 80-20%. 80-20 means हमारी कंट्री में 80% लोग एडवांस स्टेज में आते हैं, तो 20% उनका ट्रीटमेंट हो जाता है. लेकिन वेस्टर्न कंट्री में 80% अर्ली स्टेज में आते हैं, तो 20% का ही डेथ हो पाता है. So we need to increase the screening program, we need to increase the awareness program so that patients should come in the early stage in our clinic. So clinician will give the better treatment. This Marlboro man, everyone know his, you know, he got his cigarette, smoked the cigarette, then get the lung cancer. When he filed a case, so court में case हार गया था, and he put the sue the company billions or millions of dollars. Company say कि we just request you to advertise, not smoke. So this is a smoke, which very routine practice in our country. These are the different poisonous gas and chemicals which harmful for the lungs, and these are the people are doing the smoking in different way, and these are the hero. Generally we call the hero, but see what they are selling. So these are not hero. Actual fact is that. So these kind of small initiative we should take and take a action and awareness program in the country. This is a vaccine which I am talking, Gardasil, other 3 vaccine, 3 dose we need to give to the 6-year to 20-year-old girls, so that more than 99% they prevent from the cervical cancer. This is a very important slide, like 1975 to 2020. These are data in green which representing cure rate. Prostate cancer 100% curable. Breast cancer 90% curable. तो अभी मैंने क्वेश्चन पूछ रहा था कि कैंसर इज ए बैड और गुड. So see, if we come the early stage, cancer detected, so cancer patient are 100% they treated, they are healthy. Only problem that in this red mark cancer, pancreatic cancer, glioma cancer, in that researchers required. Dr. Amit Dutt is here, he working on the lung cancer, he is in the genomic level and biomarker level. You see, these are the celebrities, some celebrities which already 100% cure by taking the treatment same time. Okay, so now we need to address this problem like cancer which have the less survival incidence like pancreatic cancer, glioma less than 5%. How we address this problem? This only addressed by the researcher in the laboratory. So you see these beautiful girls, what is the difference here? क्या change है? These are monozygotic twins. Genetic makeup 100% similar. So what is the difference? Hair color. You see they have different hair color. So why it happened? If genetically similar, then what are the factors are affecting to change the hair color? Now you see this slide. This is my favorite slide. Let's eat grandpa. Let's eat grandpa. Any difference? लास्ट वाली स्लाइड का आंसर आपको यहां से मिलेगा. इसका मतलब है चलो ग्रैंडपा को खाते हैं. लाइफ ऑफ ग्रैंडपा इज इन डेंजर. बट नाउ आई पुट द कोमा. व्हाट हैपन? नाउ यू इनवाइटिंग द ग्रैंडपा. Alphabet is similar, but meaning is completely changed. In last slide, hair color, that is the reason. So this is called the epigenetics. Now, a new branch of science, without changing the DNA sequence, phenotype is changed, that is called the epigenetics. Like cancer also, genetic factor play very crucial role. Along with that, epigenetic factors also play lot of crucial role to reverse back. And epigenetic factor reversible also. Now that is in time, like DNA is your not a destiny. Your lifestyle, your thinking habit, your stress level also play very crucial role to give a shape of your genome or epigenome. These are the factors. your food habit. Okay. If you see in the Gita, they already mentioned that कि आपका फूड कैसा होगा, आप किस टाइप का खाना खाते हो, किस तरह का सोचते हो. All these factors are influence your epigenomes. If epigenome is disturbed, so your all genomic factors are changing and that impact deleterious effect on your health. So आज से आप लोगों का पिज्जा, बर्गर and cold drink बंद. So you eat a healthy food. This is the example of the bees. Okay, like female bees which eat the royal jelly, he become a queen. I am showing the importance of the food. Food are so important. These are the heroes. We take a recreational drugs into impact of recreational drug on the human health. So these are the evidence which suggest we should eat healthy, stay healthy, stress should be less. That help to maintain our epigenomes and genome which stop the deleterious effect on our health. So these are the some like advancement in the molecular biology. So we use the personalized treatment, and these are the different treatment options and drugs by the researchers developed recently. Dr. Amit Dutt and Dr. Mayank will put more light on that. I am not going the details. So these are the outcomes of modern translational research which help to treat the cancer patients, some cancer in the third stage also. Okay. So you see the incidence of ovarian cancer. The ovarian cancer is the third most leading cause of death in the female after breast and cervical cancer. The incidence of cancer is increasing in metropolitan cities. We do not know what are the factors are responsible for that. Those and high-grade epithelial cancer are relapsed in 12 months. If some lady has a high-grade ovarian cancer, within 12 months, there resistance and relapse. So no treatment options are available for the ovarian cancer, and sensitive cell become a resistant. So my laboratory basically try to understand how sensitive the patient which are in starting, so responding very well from Taxol and carboplatin, why after some time we become the resistant. So we try to understand by 3D genome architecture inside the nucleus what happened. This is a typical nucleus and the 3D genome architecture. These are defined in the different LADs and TADs, topo-related domains, lamina-associated domains. All the genes which are transcribed, they are in the nucleus, but when they are silent, like for example, tumor suppressor gene is silent and oncogene activate, so it may cause the disease like cancer. So my laboratory working on the port, prostate, ovary, testis, placenta, discovered by Ira Pastan. This family have 14 paralogs and dispersed 8 different chromosomes. They are divided into 5 groups. Group 1 and 2 come under the cancer testis antigen, means they have the immunogenic therapeutic potential. And peculiar part is that all the port members are localized on the when we divide the cancer, we classified the cancer as a clinician. High epithelial cancer is like epithelial cancer. But if you see the molecular profiling, and line 1 is a long interspersed elements, you find 2 types of phenotypes of ovarian cancer: high-grade epithelium, hypermethylated, and hypomethylated. So this is the microarray data which represent port family are overexpressed in the hypomethylated ovarian cancer patient instead of the hypermethylated ovarian cancer. When we look out their expression, ovarian cancer have the high expression of the PAN port, which stage dependent and grade dependent. So when we correlate with the methylation status, we found that the port family are highly expressed in the hypomethylated patient as compared to the hypermethylated. It indicates that methylation will play a very crucial role in the port expression. So epigenetics role is there. And finally, you see here, the laminopathies like LADs are play crucial role. If any defect in the LADs, it may cause the different health problems, cancer, neuropathological problems. So here we try to look out the lamins expression in the different cancer, and we found that lamins are play very crucial role in the signaling pathway of the cancer. And finally, We have published this paper, and you see RNA-seq data. We found like TCS data and geo data where we found that lamin expression in the ovarian cancer patients. And you see, after that, we choose the model, like in which model we should experiment to understand the lamina domain. So different ovarian cancer cell line, we see the expression. So 82780 cell line we have choose because there are differential change in the lamins and port expression. And after that, we confirm it. And we did the LCMS to look out what are the crucial pathway play in the roles in the port regulation with respect to lamin change. So along with that, we make a cisplatin resistant cell line, which is a fourfold is resistant with respect to the wild type cell lines. After confirming the models, we start mapping the LADs, lamina acidosis domain. Here what we use the DAM. DAM is a DNA adenine methyl transferase enzyme, play immunogenic role in the bacteria when bacteriophage infected, so it help to self and non-self. Okay, and bacteriophage genetic material chopped out. So here we use that DAM clone with the lamin B and transfected in the both the cell line, and then use Oxford nano platform and Illumina platform for sequencing the LADs. So once we use the bioinformatic pipelines, and you see twofold up regulated is the showing the interaction, and negative is the no interaction with the port family with the LADs. So here we, after performing the sequencing, we use bioinformatic pipelines, and I am showing the representative port members which are interacting here. All these are port members which are interacting. And similarly, after mapping this map, you see LADs. LADs in the sensitive cell line is big and less interactive. Once in the sensitive, in the cisplatin resistant cell line, the LAD size is small and interaction also go less. So these are the factors which may help crucial role in making a sensitive cell line to the resistant cell line. Once along with that, we know repressive and active marks. These are play very crucial role in the regulation of the gene expression. So we look out the immunofluorescence experiment, then did the native chip experiment to map out the expression of lamin A, because A and B play very crucial role in construction of the LADs. So the occupancy graph, I am not going in details of that. These are interactive pictures of the IGV which show the interaction of the port with the LADs. And you see here methylation marks, active and repressive marks, how they make a resistant cell line, sensitive cell line to become a resistance. There are different port members, their interactions. And finally, as this data suggests that port family are regulated through the epigenetic mechanism, and I also said that port family is a immunogenic. So could we make this protein any kind of vaccine? So our hypothesis that the hypermethylated patient, we have less port expression. Could we treat the epigenetic modulated drug and increase the expression of port and make a DC-based vaccine against that? So in that hypothesis that after we recruit the patients, take their blood samples and look out their expression of lamins and port expression. Here you see the Western country have the negative correlation, but our Indian patient cohort show the positive correlation with the port expression. After native chip, we look out their different expression, and we treat these cell lines with different epigenetic modulated drug. You see decitabine increase around 3.5-fold of the port expression, and decitabine is a FDA approval drug routinely used in the clinical setting. After that, we use the two peptides which are immunogenic and co-culture the OCAR cell and make a DC-based vaccine. And we see the caspase activity assay, the cell line which treated all the epigenetic modulated drug, highest cell death we found there. So this is the proof of the concept. On the basis of this proof of the concept, we are now under the process to conduct phase 1 and 2 clinical trial on port-based DC vaccines. So that is the under process. Along with this is the laboratory work we have done. Like Dr. Amit, Dr. Mank, me, and lot of other people are working day and night in the laboratory, and clinicians are working in the clinic. But our age, you know, our population size very high. So along with that, we need an infrastructure also. So I would like to say, Professor Rugg, to develop NCI Jhajjar campus, which is just a small movie in the night campus. You see, there are 300-acre lands, 710 beds, dedicated OPD, research block, here academic block are there. This is a small video of campus. Now this campus actively working, and with time it is progress well. All the AIMS faculty and ISS faculty especially working day and night to make this campus more active. So I would like to say thanks now my funding agencies, my students for providing this fund, and this is my lab pictures. So like time to time they get some awards. All the work which I have shown here, they're due to my students. Credits go all my students. And same time we visited timely every year. And this is the small video of my lab visit, Jojila Pass, 0 point. So all my lab members are visited there, and like along with my research, we also have some fun. So now I will say thank you and looking forward to your questions and your suggestions. Thank you very much.
Thank you, Dr. Sharma, for giving a hope that cancer can be cured. Definitely your observations have added a great depth and clarity to the deliberation. Once again, a big hand for Dr. Ashok Sharma, ladies and gentlemen. We now come to one of the most important segments of today's program, and that is the signing of Memorandum of Agreement between GSBTM and BCIL. GSBTM is facilitating the development of biotechnology in the state and providing financial assistance to academia and research institutions under research support scheme to undertake research on the problems relevant to the state and their solutions through biotechnology interventions leading to product, prototype, and technology development. GSBTM has partnered with Biotech Consortium India Limited, BCIL, which is a public limited company of Department of Biotechnology, Government of India, with a long-standing expertise in IP management and technology transfer in biotechnology to facilitate IP protection and commercialization of research outcomes. May I request Shri D.D. Jadeja, Mission Director, GSBTM, and Dr. Purnima Sharma, Managing Director, BCIL, to kindly proceed for the MOA signing. This is a historical moment, ladies and gentlemen. Can we have a huge round of applause for this? This Memorandum of Agreement between Biotechnology Consortium India Limited, BCIL, and the Gujarat State Biotechnology Mission, GSBTM, formalizes a strategic partnership to jointly strengthen biotechnology innovation, capacity building, and technology commercialization. It brings together BCIL's national-level expertise and GSBTM's strong state-level research ecosystem to create new opportunities for startups and biotech industry in Gujarat. Once again, give a huge round of applause, ladies and gentlemen. This MOA exchange marks a significant milestone in strengthening collaboration between academia, government, and industry, and we are confident that it will pave the way for impactful translational outcomes in cancer research. We thank both organizations for taking this important step towards shared progress and innovation. At this monumental moment, it is now my honor to invite Dr. Purnima Sharma, Managing Director, BCIL, to deliver her remarks on From Discovery to Deployment, Accelerating Translation of Cancer Innovations. Dr. Purnima Sharma is the Managing Director of Biotech Consortium India Limited, BCIL, leading national efforts in biotechnology commercialization, IP management, and technology transfer. She holds a PhD in experimental medicine and completed her postdoctoral research at IIT Bombay and has traversed through diverse roles within BCIL over more than 3 decades, culminating in her current leadership position as a recognized expert in translating biotech innovations from lab to market. Madam, we request you to address the audience, please.
Honorable Shri Digvijay Singh Deja ji, Dr. Ashok Sharma, Dr. Ansuya Bhadalkar, distinguished experts in the audience, and dear participants. In fact, I would like to express my heartfelt gratitude to GSPTM and especially the visionary leadership of Shri Digvijay Singh Deja ji for having recognized the importance of transfer of technologies. And that's why this transfer ceremony has happened. So I'm extremely grateful, extend my thanks to you, sir. So in fact, today's presentation is also in the same direction. So can I have so the first presentation? Oh, okay, he's here. I'll do it myself. So just before that, I'll just briefly inform you about my company. Pardon me if I stand there, I won't be visible, so I thought I'll come here. So I hope I'm audible. Thank you. So this is a company which was set up way back in the year 1990 by the Department of Biotechnology, Government of India, to act as a bridge between research and the industry for facilitating commercialization of biotechnology. So we have a very vibrant and very elite board of directors which have heads of all the government departments, whether it's DST, ICMR, ICAR, all the science ministries which are dealing with biotechnology development. And the chairperson is Shri Shekhar Mande, who's the former DG CSIR. The key areas of operations, as I said, is technology transfer. Management of IP becomes a part of tech transfer because biotechnology being a highly knowledge-intensive area, unless the IPs are managed, we can't think of technology transfer. It's also a highly regulated area, so the regulatory support, consultancy, because market assessments, all that comes under consultancy. So we have, transferred more than 85 technologies in the various disciplines, including diagnostic, therapeutics, drugs, etc. Extensive experience and expertise in this area. Consultancy, of course, as I said, no tech transfer happens unless we have, idea about what is the market potential of the technology and the products we want to commercialize. But also it is important that we should have expertise in preparing DPRs and feasibility studies, and also assisting, industries when they take the technology forward in terms of regulatory support, in identifying the gap areas so that they can be bridged, investment decisions, market analysis, etc. So because we have different verticals, whether regulatory or consultancy, etc. But in the area of technology transfer, the main areas that we would be addressing, and again taking in the context of today's MOU, these are the areas that we would like to extend to GSBTM2: IP management, technology transfer, collaborations. No tech transfer can happen without effective collaborations. sensitization and entrepreneurship. So today is the time of spin-offs and startups, so it's very important to have entrepreneurship promotion also important. So in the context of today's conclave, how can a TTO help, in taking the cancer innovation forwards? So as we all know, and from the presentation we just saw of Dr. Ashok Sharma, cancer research is witnessing high rate advancements, extraordinary advancements in terms of novel therapeutic biomarkers, the cutting edge diagnostics, therapies, and AI-driven, artificial intelligence-driven decision tools. However, we had extensive discussions yesterday among the experts. We have here Dr. Singh and Dr. Dutt. We know that many of these technologies, they don't reach the end users, the patients. And that is because the journey from laboratory to the real world deployments is very complex. And it is also, there are a lot of uncertainties along the path. And therefore, TTOs can play a very enabling role in bridging this critical gap. So now let us look at, why the translation gap is there. What are the reasons? The main reason is because the research in all these, institutions are primarily driven by academic curiosity and not from the standpoint of the clinical adoption. I'm not saying always, but largely it happens so. And it is true because, the academic curiosity has to be there. The passion should be there to take it forward. But at the same time, applicability of those research is equally important. And also, accordingly, because it is driven by academic curiosity, the validation in the real life settings doesn't happen. And that is in the clinical settings. Of course, I know a lot of experts who are here today also, including Dr. Singh, who are working, and also Dr. Sharma, who are working in great institutions like AIIMS, and they know the importance of validation in clinical settings, and they're doing that. But people who don't have connects with such hospitals and clinical settings, they are totally unaware of the importance. And this is what we have seen when we are transferring technologies which have high implications in healthcare sector, but then they are totally unaware of importance of testing them on patients. There's weak understanding about the regulatory pathways. So although some data is generated, but then it is not in conformity with what is required by the regulatory agencies. So this is no use then for the industry. They are unaware of the market pathways. Now, who is going to be the end user? Is the diagnostic labs, or it is the hospitals, or it is some other intermediary? And also the IP strategies. Sometimes they just go and publish it, and it is too late for the IP to be protected through patents or other pathways, and therefore the industry don't find it lucrative. Or even if the IP is protected, it is not protected in the right jurisdictions and where the market is there. So it is important to know what are the countries, what are the jurisdictions where there's a market for this particular the technology, and accordingly that protection has to happen at the right time. And also because there's limited engagement with the industry and the end users, so it does not it's not it doesn't use its maximum potential to the potential to which it can be exploited doesn't happen. So therefore, what happens is that promising cancer technology, they remain stuck at low technology readiness level, whereas the huge potential that they are embedded in them, they could have been exploited if the right guidance was provided to them by effective TTOs. So what can a TTO do? They can act as a translation partner. But by that I don't mean that they will effectively file the IPs. No, but what they can do is they act as a de-risking engine. So they will reduce the uncertainty, and how do they do is they assess the technology not only from the technical standpoint, from the IP strategy point, from the regulatory strategy and the market, put together all this information in a format so that industry or the startup or the spin-off can have a look at this and then appreciate the potential of the technology and then take a decision to take it forward. They can also act as a connector between the researchers, the clinicians, the hospitals, the industry, so that all the gaps which are there in between can be bridged. Because the people who are actually going to take it forward to the market, they better understand, and if engaged with them early on, we'll be able to know what are the gap areas, and mitigation strategies can be taken early. So therefore, the interventions are early assessment of clinical and commercial applications, strategic planning of the intellectual property and then protecting them, also facilitating co-development, because it is not that we develop a technology and then look for an industry. It may be too late then. So mostly many a time co-development, especially in case of cancer technologies, it is important that we look for co-development strategies, and also enabling startups and spin-offs. So yesterday also we were looking at another technology developed by another institutions in Ahmedabad, and we thought that co-development would have been a better strategy rather than developing it and then going forward. So all these co-development models are important, and especially in case of cancer technologies, we should look at them proactively. So it's also important when we're looking at deployment of cancer technologies in low and medium the settings, income settings. So it is important that the technologies should also address important points such as accessibility, affordability, and scalability. Because it should not be that they are only for some elite few. It should be able to address the requirements of the people who are from low and middle class settings, and therefore it should be frugal. It should the regulatory and the validations should be simplified. Partnerships with public health systems, and also the licensing should be responsible. So we should not be that very high licensing fee, very high royalty, so that the cost actually goes up and the actual cost to the patient becomes very high. So all these are equally important, and unless all these important components are taken care, we can't expect the technology to reach the patient, especially the poor patients. And therefore, to accelerate transitions, what the researchers should do is to engage with the technology transfer offices of the respective institutions early enough. Don't go and hurriedly publish it, because publication can be done anyway after it is patented. So think beyond novelty and usability to usability and impact. So impact is more important. I agree novelty is important, but since looking at the wide incidence of cancer which is happening in our country, I think all those who are engaging in research should also start looking at the impact that the research that they are doing it, and also collaborate with clinicians and industry during the research design itself, so that it should not happen that we develop something and later on the industry feels that it should have been designed separately or differently. And we should be open to co-development with the industry and not develop it and then find a licensing partner. And we view IP not an obstacle, because normally scientists, they have a tendency that I must publish it and I should publish it fast. But then in the process, the commercialization becomes a difficulty. So IP is not an obstacle, but actually it is an enabler for the impact. And therefore, translation is not a compromise to the science, it is just an extension. So this is how the scientist must view it. So to conclude, the future of the cancer care depends on not just what we discover, but how quickly it is deployed responsibly. We should also align the research, excellence, clinical relevance, IP, all this should be aligned together. They should not be, silos. Research is one silo and commercialization another. So everything should be, work as a combined ecosystem, so that the deployment happens fast. And the TTOs like us, we are there to help and stand by you as co-creators in this journey to ensure that the cancer innovations reach the patients. Thank you very much.
Thank you so much, ma'am, for your enlightening and encouraging address. We are grateful for your leadership and continued support. May I now request Dr. Ansuya Bhadalkar, Joint Director R&D GSBTM, to propose the vote of thanks for the inaugural session.
Thank you, Manali. Good morning, everyone. And as you can see, I'm very privileged to be standing here proposing the vote of thanks. And I hope, unlike ma'am said, that I am visible. I hope so. So I'll not change the mics. So when we started planning this conclave, almost 2 months back, we started the planning. So we were visiting in the entire database of all the research that has been funded by GSBTM, and I am a new joinee into the R&D pillar. So my team was there who were old and they were working in this space. So they actually informed me that the highest number of projects that have been funded in a specific sector is cancer. So we said that then we need to listen to those people who have done this work and see if we can, collate the work and if we can bring out something sensible for the society from all the research that we have funded. So ये सारा plan उठा के हम हमारे मिशन डायरेक्टर सर जडेजा सर के पास गए और हमने कहा कि सर हमको ये प्रोग्राम करने जैसा लगता है. The only thing, there were 2 sentences he told me, and उसके बाद भी सर ने बहुत ज़्यादा कुछ नहीं बोला. उन्होंने सिर्फ ये बोला, करो, अच्छा करो, और इसमें से कुछ निकल के आए, आगे का कोई रास्ता मिले, ऐसा करो. That is all I want from you. So that is how encouraging he has been with all the activities that we have been conceptualizing at Mission. So I thank our Mission Director, Sir, D.D. Jadeja, Sir, for being so kind and so supportive. In the same league, in the same breath, thank you. In the same breath, I will also want to thank our Secretary, Department of Science and Technology, Ms. P. Bharti. Although she is not here, in her absentia, I want to thank her for the very quick review and approval that she gave for the agreement that we signed with BCIL today. It would have not been possible if she would have not been that quick and she would have not been so positive. She took just one meeting and she said, yes, I want this to happen, and you ensure, she told me that you ensure that this further translates into something that is givable to the society. So I am hoping that with this agreement with BCIL, we will be able to push this agenda further, and that is what, as a funding agency, we always want. So I thank P. Bharti ma'am, Secretary DAST, for being so supportive. I will thank Dr. Purnima Sharma for bringing up this idea, this thought of having an agreement in this space of tech transfer, because she has been a scientist at one time, and now she is on the other side of science, and that is administrating science, and she has seen over the years how difficult it is to take the research from a laboratory and translate it and make it a usable technology or a usable product or a usable process. So she has been working in this space, and she came up with this thought that let us have an agreement and let BCIL help the state researchers to make a sense out of their work. So thank you, ma'am, for bringing it up, visiting us, and making this day a possibility. I thank Dr. Ashok Sharma for sharing such wonderful insights into how cancer is so varied and what are the statistics of our country. Because statistics change with every country, and today while sitting through his talk, I got to know that ovarian cancer is the 3rd highest killer amongst all the other cancers. So I think, yes, it is a time that we need to wake up. It is a wake-up call for all of us. The vaccination that he talked about is a very important aspect of dealing with cancer. At least there is one cancer for which we have a vaccine. So I think as a platform of awareness generation, we all should take home this message from here that if there are vaccines that are available, we need to go ahead and have them so that we can protect ourselves and our next generation of female child that we have. So thank you, sir, for emphasizing on how important vaccination is. Certainly we will, on personal discussions, we will want to know more from you as to what as a state we can do to have a larger network of cancer research here, and how we can use this research work, club it with the national data and see what sense we can make out. That will give us a demographic idea of the demographic spread of the cancer that is happening in our state. Thank you very much, sir. I will also want to thank all our speakers who are right now not on the dais but will be talking to us throughout the day. I thank them for sparing this kind of a time, spending a whole day with us. All of them are big shot researchers in their own fields. So sparing the time and sharing their insights with us so that we as a state funding agency can pick up some of these insights and do something that is worthwhile and important for our state. I thank all our speakers. Nevertheless, I will also want to thank all our participants, because none of the participants here are students. They are all faculties, researchers who are doing great work in their own domains related to cancer or related to the tools of biotechnology. They have pulled out a whole day to be a part of this program so that they can orient their research and do something more useful with their research. So I thank all the participants for being here. And last but never the least, my very dear team, because they have put in relentless amount of hours and thoughts into this entire program. Whole of the arrangements that you see here today are done only by them. So I have not been a part of it. I have just been coordinating with all of them, and they have coordinated it further. So thank you, dear team, for being so very supportive for this work. And I cannot close my thanking note without thanking Professor Rakesh Rawal, Gujarat Biotechnology University, because he has been a powerhouse of knowledge and support, not only to the program, to all the initiatives of GSBTM, but especially for this program, because he has enormously helped us curate this program so that we can have a confluence of national and state data into this conclave, so that we can make some sense out of the whole work that has happened in the nation and in the state and take it ahead as a funding agency. So thank you, Professor Rawal, and thank you everyone for joining us. A heartfelt gratitude to all the people whom I have not mentioned here, but they have been a cog in the entire machinery. So thank you, everyone. Thank you.
And we thank Dr. Ansuya for the guiding force of this entire conclave that she is. A huge round of applause for Dr. Ansuya, ladies and gentlemen. All right, so now it's time for a tea break. We will commence the next session in just 15 minutes. Thank you so much. Hello. May I request all the speakers of the day to come on the stage for a group photograph? I once again request all the speakers and entire GSBTM team to gather near the stage for a group photograph. Once again, I request all the speakers to come up on stage for a group photograph, and I request the GSBTM team as well. All the speakers are requested to come up on a stage for a group photograph. All right, ladies and gentlemen, I request you all to be seated. We are resuming the session now. People who are standing are requested to take their seats as we are resuming the conclave. Those who are standing outside, if my voice is reaching you, I request you all to come inside and be seated. Welcome back, ladies and gentlemen. We are now resuming the conclave with the session on National Research Scenario. The session aspires to capture a glimpse of the different aspects of cancer research and how technologies are influencing ease of diagnostics and treatment in cancer. To start the session, may I request Dr. Amit Dutt, Professor at the University of Delhi, to share his perspective on the topic, Translational Cancer Genomics from Molecular Insights to Clinical Impact. Dr. Amit Dutt, a Shanti Swarup Bhatnagar awardee and a professor at the University of Delhi, formerly Scientist G. Actrek, is a leading cancer geneticist specializing in the somatic landscape of Indian-specific malignancies using next-generation sequencing and functional genomics. His translational research identifies novel mutation and therapeutic targets in gallbladder, head and neck, and early onset breast cancers to advance precision medicine and biomarker development. May I request Dr. Amit Dutt to kindly take the stage. Ladies and gentlemen, with a huge round of applause, we'll welcome Dr. Amit Dutt on stage.
So, a very good afternoon, everyone. Thank you to the organizers, especially Dr. Pankti, for having me here today. So, So as I have already been introduced, I'm coming from the Department of Genetics, University of Delhi South Campus. However, I have relocated there very recently, last year. So the work that I'm going to be talking about today was primarily carried out at my former institute, that is ACTRAC Tata Memorial Center, Mumbai. And today, I will be restricting my talk primarily to the lung cancer. Even though my laboratory is involved in focusing in other different cancers, such as head and neck cancer, breast cancer, cervical cancer, as well as gallbladder cancer that has just been kind of like mentioned. We usually take genomic approaches because we consider ourselves to be dumb people. We do not have any hypothesis to begin with. We go in a completely unbiased way. We look at the genomic alterations that underlies the cancer genome, and after statistically, in a disciplined manner, analyzing the data, what we do is that we generate hypotheses. And when those hypotheses have been generated in a completely unbiased way, using genome-wide approaches, we take functional approaches to falsify those hypotheses. When those hypotheses could not be falsified, they stand the test of the rigor of the experiments. Those hypotheses kind of like allows us to move further. So to begin with, I wanted to give you a glimpse. Not exactly sure if my slide would be totally visible. I wanted to give a glimpse of how the cancer incidence across all different cancer types globally, as well as how does it really align to the Indian incidence. In general, as what Dr. Ashok has already kind of like specified in his presentation, the incidence of cancer in India is relatively about 1/3 as what you kind of like get to see in the countries with a higher income group. That's by the number every 100,000. However, as what Dr. Sharma has also kind of like specified, that given that the absolute number that what we have, the denominator kind of like goes very high. It would be somewhere around, if you would be comparing that somewhere 98.5 or like close to 100 per 100,000 in the Indian. So we may have lower incidence of cancer. However, what is more astounding and what is more striking is that we are more likely to die of the disease. As what you would see in the first row, and if you would compare it with the second row, and if you would see as the age-adjusted mortality which is associated, despite having 98 or around close to 100 as an incidence, about 65 of them are dying. And now this, if you would compare to the global or to the US standards, there the death is kind of relatively much lower. Coming to specifically to Gujarat, I was just kind of like trying to look through it. It is something which is at par with the national standards. However, there appears to be that the incidence of few cancers in general, which kind of like skews the data. It appears that the incidence of cancer in Gujarat is slightly higher as the national standards. And this is primarily due to the smokeless tobacco chewing habit that drives it. Those are primarily the head and neck cancer, the oral cancer. When it comes down to the lung cancer in particular, as what is shown in the lower panel, the trend around remains the same. And you can see how deadly the disease is. If you would look at the mortality or the incidence, if it is 5.8 in the Indian setting, as what I mentioned earlier, we are more likely to die of the disease. If you look at the mortality, it is 5.3. Now, this is striking as you would compare with the other nations, and in fact, of late, Dr. Ashok was mentioning that infectious disease and the deaths or the cardiovascular diseases, the deaths, those are associated with this are at a set of trend to decline. The cancer deaths associated, especially in the countries with the lower income groups such as India, has hardly there been any change over a period of last 5 or 6 decades, which kind of like brings a very grim situation. Be it so initiative to understand these cancer, which are more predominant in the Indian subcontinent, the initiative has to have to taken from the Indian setting. It appears that this might be a disease in its own setting, which might be slightly distinct. So my laboratory, over the period of last 14 years at ACTRAC, Tata Memorial Center, along with the clinical friends of Mars, primarily in lung cancer, we have been collaborating with Dr. Kumar Prabhash, and along with them, what we have done over a period of time is that we have characterized all the major cancers, major subtypes of lung cancers. We have been able to describe all the baseline reports across that what are the major players which underlies lung adenocarcinomas or different subtypes of lung cancer. And we have found some many surprises out over there, such as what we know is that the major players across the globe appear to be the same. End of the day, we are still Homo sapiens, right? So the genes which are altered or underlies lung cancer, as what you kind of get to see in the Caucasian population, those genes are altered in the Indian lung cancer patients as well. However, what we tend to get to see that it happens at an altered frequency. For example, EGFR, that is an epidermal growth factor, it's a tyrosine kinase, is mutated at a frequency of 10 to 12% in the Caucasian population and is known to be altered at a frequency of 35 and higher in the East Asian population. We were one of the first ones to report that in the Indian subcontinent, this frequency is intermediate. And when it comes down to the KRAS, there is a completely opposite trend. We see that the frequency of the KRAS is lower in the Indian subcontinent as compared to the other ones. We went on to characterize newer alterations as well that we think that are primarily afflicting Indian subcontinent, and we made discoveries such as other tyrosine kinase as FGFR3 and all, but that's not specifically what I'm going to be talking about today. What I thought is that you are very well aware of that most of the, especially in the lung cancer, most of the therapeutic targets and the therapeutic options which are designed against those therapeutically relevant targets have really revolutionized the cancer treatment, and specifically in lung cancer. Should I be speaking to you in 2004, the diagnosis of a stage 4 lung cancer would be a death sentence. A patient would not be surviving beyond 4 to 5 or 6 months max. Today, that patient, thanks to all the discoveries and all the understanding of the underpinnings of the genetic alterations, if they are positive or if they are harboring any of these known therapeutically relevant alterations, you may easily extend the life for at least 30 months more, and if they respond to immunotherapy, you may imagine that those patients might even survive for more than 60 months. Such is the drastic and significant impact. We are comparing from 4 to 5 months to 60 months, depending on the kind of alterations that they may underlie. However, there comes the caveat. There is invariably a resistance that kind of develops. These patients, when they are given personalized treatment that is directed primarily against specific alterations, they are more often than not likely to come back with the progression of the disease. And then there are different generation of drugs which are usually offered to the patients. A student in my laboratory, Supriya Hayat, wanted to understand, and before her, Asim Joshi, wanted to understand the different mechanisms that were underlying the lung cancer resistance. So what they were essentially wanting to do is something that I want to kind of talk about in the next few slides. To begin with, what we know about the lung cancer resistance, there are upfront, primarily for the students, there are two different kinds. Kind A is those patients who are upfront resistant. They are intrinsically resistant despite they being positive for the alterations which are therapeutically relevant. They might still not respond to the treatment, and you would see that within few months the disease would kind of come back. However, more often than not, what you tend to get to see is an initial response, and once they have initially responded, within a few period of time, they again kind of come back. So there comes the question, a biologically relevant question, that what are the relevant mechanisms that underlies it? What we do know from the textbooks, it is already kind of like well characterized now. We know that there are different kind of genetic alterations that may underlie resistance. It can be a mutation, a second mutation in a gene such as the EGFR, which is called as the T790M, and other T790M-like mutations which may give resistance. B is that there could be a second alteration in the same pathway as what is shown, such as the mutation in KRAS or other genes in that pathway that again make those patients resistant to the treatment that is being targeted against the EGFR. In addition to those, there could be a bypass mechanism where there could be a parallel signaling pathway that may be altered and that may be activated. So now, even if you get rid of the EGFR signaling pathway, the patients would still the disease would still kind of like progress. In addition to that, it is well documented in literature that there could be a histological transformation, as what is shown on the extreme right, where there is a transformation of one cell type to another cell type. So Supriya was interested to understand, and Awesome Joshi was interested to understand what is kind of like something more that we could learn from the Indian patients. And along with Dr. Kumar Prabhash, we went ahead and characterizing that. One of the things that really that this project started from is this molecular report. This is a molecular report where essentially a clinician asks a patient to get a molecular diagnosis done for the genomic alterations that it may lie. And if you are sitting in a molecular tumor board along with the clinicians, this is the report that would come in front of you in the table, and you would see that these patients are positive for oncogenic alterations such as EGFR. What would be the default action? The default would be that this patient should be treated with osimertinib or darcitinib, depending on the generation of inhibitors, that would be kind of like offered to the patient because there is a presence of EGFR alteration. What didn't escape the curious eyes of Supriya Hayat in the lab is that there are other alterations as well. What are those other alterations? You could see that there are tumor suppressor genes. In addition to the EGFR, they are also mutated. But in a molecular tumor board, we often tend to overlook those alterations which are in the tumor suppressor genes. And why does that happen? That happens because there is no treatment against those tumor suppressor genes. Despite they being present over there, they need to be ignored. So Supriya asked a question. The question is, what are these co-occurring alterations that is happening along with the oncogenic alterations as what you kind of like see over here? My apologies that those slides are not very visible because of the distance. Essentially what it is reading is that on a row 1, there would be an oncogenic alterations in EGFR. Row 2, 3, 4 are alterations which are co-occurring in the same patient. And this is you tend to get to see different med-oncs sitting over here. They would kind of like met for it that this is something that is a typical report that you kind of like get to see. So what did Supriya do? Those are the ones which are the co-occurring along with the EGFR. So what did Supriya do to start with an experiment, a basic experiment? She goes into the public resource data set, and that is the Cancer Genome Atlas, which is the compendium of all the different, about like more than a dozen publicly funded genetic profile of those cancers. And there, what she does is that she takes two bags. Bag A, in which she puts EGFR alteration patients, and bag B, in which EGFR, along with other tumor suppressor genes which are altered. And when she compares, she finds a very interesting finding. She finds that those patients where there is a co-occurring alterations, they have a much more adverse survival, which is whether it is an overall survival or whether it is a progression-free survival with a significant hazard ratio associated with it. Which brings that, okay, that means these tumor suppressor genes which are happening along with the EGFR, they're doing something which is relevant and significant that makes a worth biological question to address. So what does Supriya do? So then, along with Dr. Kumar Prabhash, what we did is that we sit on the table and we decide that we need to systematically address this. Now, that being the strength of the Tata Memorial Center, that we get the rarest of the rare sample. And that's what we do in this case. Along with Dr. Kumar Prabhash, we collect 3 different samples from the same patient. At the baseline, a biopsy is taken, or maybe an FFP block is from the patient, and then also the blood, which is from the same patient, and along with that, the patient is offered a treatment, and then the patient is longitudinally followed up. When there is a progression and when the patient again the disease relapses, you again take the same biopsy from that patient, and now this way there are 26 patients shown over here where they are categorized by Supriya in 3 different types based on the literature: early relapse, late relapse, depending on the duration of the relapse, and then we go ahead and we perform the classical whole exome sequencing across these samples at a high depth coverage. What we find is something interesting. Those are not very much visible over here. Supriya wanted to understand, the question that she wanted to ask was that mutations, those were present in the baseline at the time when the patient presented to the clinician, and the mutations when the disease relapsed, say after even 20 months, which of the mutations, which of the genes which were harboring those mutations, they were retained. Because there was a significant time period that that had taken for the disease to kind of like relapse, which would kind of like suggest, okay, these mutations are something which was important for the survival of the tumors, and that's why the tumors were retaining those alterations. And when she when she did that, what she found is something very interesting. She found that there were enrichment of those genes which were primarily of tumor suppressor genes. Now, this is something consistent with what Supriya had already hypothesized looking at the molecular reports. So, we went ahead and we kind of like went ahead with the further investigations into this. However, I will come back to this slide. I will come back to this whole exome sequencing and talk about some other aspect associated with that as well. It was not only that these mutations that they were retained. That is, these mutations were present on the baseline on day 1 when the patient presented to the clinician, and were retained after 20 months. Even their allele fraction was increased. What does an allele fraction means? It means that if there is 100 cell, of these 100 cell, initially it would be the 5 cell that were kind of like harboring the alterations, and now it is 20 cells. So that way the allele fraction would be increasing from 5 to 20. However, we challenged this dogma, and that I'll come to like later on as well. There was an increase in the allele fraction in the EGFR, in the P53, and in tumor suppressor genes in general. So these were all pointing towards that the co-occurrence of these tumor suppressor gene were more than by chance, and there appears to be a systematic biology that might be underlying. So what do? We approach our friends in Pune, One Cell Diagnostics. And along with the One Cell Diagnostics, what we do is that we perform, because we have now the longitudinal collection of samples of the patients since the early detection, of early diagnosis and the relapse, and we go ahead and we use their proprietary comprehensive genomic profiling panel, which is called as OncoIndex, and which has about like 600 odd genes to be like ordered, that could be investigated. So we perform, we take the ctDNA, we perform liquid biopsy on these guys, and these are the points where the collections were made. In total, about 243 comprehensive genomic profiling were performed, and they were analyzed. So all the analysis, not taking you into the details because that's not exactly the focus, all the analysis in my laboratory is done in-house. We do not outsource our analysis. First thing first, to the students, whenever you are embarking in any of these kind of like projects, you need to be sure that where you're heading to. And one and the first and the most important thing is the positive control. So what is shown over here is the upper panel is the set of mutations that was there based on the whole exome sequencing of the primary tumor, and the lower panel is by the liquid biopsy from the blood. So the question is that if there is a concordance between the primary tumor, that is the tissue biopsy, and the liquid biopsy. And we found that yes, those were highly concordant. Now, that's not rocket science because that has been kind of like well established in the field, but then at least that tells us that we are on the right track. And then what do? we kind of like take these samples, and this is one of the example that I'm kind of like showing you around for the patient where there is an early relapse. And we go ahead and we perform clonal analysis. Essentially, not taking you into the details again, what underlies it, in the clonal analysis, because we have now the statistical power of 243 samples, and the samples which have been longitudinally collected over a period of time, we wanted to see how the tumors are changing or how they are evolving. And what is kind of like shown over here is a very interesting fact. You would see that there is a clone. How is a clone defined? Clone is defined based on the mutations and based on the copy number alterations that it kind of like harbors, in brief. So you would see that in center there is a red clone that is coming up. That red clone at the time point where it kind of like starts up, that is the clone which is kind of like driven by a known resistant mutation, that is the T790M. Now, if liquid biopsy is performed, this emergence of this clone and of this mutation could be detected much early on, so that would give an arm to the clinicians to know that, okay, this patient is likely to develop the resistance much early on. Be it so. So this is how it kind of like really looks like, the clonal analysis. The clonal analysis of patients which are in early relapse, mid-relapse, or the late relapse. What is most important is that the focus that I want to draw your attention to, that Supriya was primarily interested, if you would recall, that she was interested in identifying those genes or mutations which were there and the day one, and those mutations that were retained after the relapse. So there is a clone which is called as the founder clone. The founder clone which was there at the beginning, and that's those clones exist till the end. When she looked at those founder clones, which is the one which is shown around, I'm not sure, I have a small problem with the color, what color you are seeing. I think it's a purple, I don't know, the first clone. Students always, they make red color things which I'm not able to see properly. So essentially what we found in the foundation clone is that there was more of an enrichment of tumor suppressor alterations, tumor suppressor genes which are kind of like altered. Now this is an independent validation on a liquid biopsy. We are again and again finding alterations which is there on the molecular report, that in the foundation clone there is more and more of a tumor suppressor gene. Statistically to present, this is how it kind of like looks. The tumor suppressor genes are more often altered in the early, intermediate, and the late. Now, this is super interesting. And if this data is present to the clinician on day one, then the clinician becomes empowered to predict or to have that prognosis in there, that here is a patient who are more likely to relapse based on the co-occurring tumor suppressor gene alterations. That's what the statistics is kind of like telling us. This allowed us to identify a set of like 17 and who are the patients who will be receiving a single agent therapy based on the screening that we have been listening to the information. So based on this study, we were able to identify or stratify those patients who are having co-occurring alterations. still there and the patients respond to it. Be it so, that brings down the cost by 1/7. But be it so, I want to just kind of like thank you to all the students. I have primarily presented work today which has been performed by Supriya and Aniket and Sanket in the laboratory. I have a former member of my lab, Dr. Rohan Chaubal, also sitting around over here in the audience, who had earlier worked on breast cancer in the lab. That's what we have continued. This work was done by with Dr. Kumar Prakash. My laboratory has been very generously funded by the DBT, by the Tata Memorial Center, by the DBT VNCI, by the Terry Fox Foundation, more recently by the ICMR and the Tata Innovation thing. Thank you for your help, and I shall be happy should there be time for more questions.
Thank you so much, sir. We sincerely appreciate your valuable insight and delivered with excellent, remarkable clarity and commanding yet composed voice that held everyone's attention, I believe. Thank you so much, sir. So now I'll request Dr. Nitin Dave to come up on stage and present memento to Dr. Dutt. Can we have a huge round of applause, ladies and gentlemen? Thank you so much, sir. May I now have an honor to invite Dr. Pahini Pandya, founder and CEO, Panakia, to share valuable insights on the topic AI-based cancer diagnostics. Dr. Pahini Pandya is the founder and CEO of Panakia, an AI-driven diagnostic company, a former cancer researcher with a PhD from King's College London. Her company develops technology to provide rapid cancer profiling directly from tissue images, aiming to significantly shorten diagnostic times without the need for additional lab tests. May I request Dr. Pahini Pandya to kindly take the stage. Ladies and gentlemen, come on. Let's give a huge round of applause for her. Thank you.
Hi, everyone. Before I begin, I want you to imagine you have a cancer scare and you've been waiting for more than a month for results of many different lab tests. My presentation is not showing. Brilliant. So when doing a cancer-focused PhD, that was me. I had to wait for more than a month for results of many different lab tests, so molecular assays, in order to get an all clear. I was lucky to not have cancer, but many other patients in that waiting room were not. And that was the moment when I thought, if this is the situation here in the UK, it's much worse in other parts of the world. And that's why, aligned with Dr. Purnima's point, I founded Panakia with the vision of making precision medicine accessible. When I say accessible, it's not just cost, it needs to be timely. It needs to be accessible to a wide range of people without need for high-end capital infrastructure or very specialist expertise. And it also needs to take into account limitations or technical limitations around biology, including limited tissue availability. And that's what we are addressing at Panakia today. So it is a well-known fact, and this audience needs no introduction on how critical understanding biology is to developing the right sort of drugs and treating patients with the right sort of treatments in the clinical setting. And despite billions of rupees being invested in understanding biology at a molecular scale, so your DNA, RNA, protein, metabolite-related interactions, or multi-omics, drugs still fail in the clinic. And that's because missing information is still the largest, biggest challenge in biology. And that's because most of these multi-omic assays consume tissue, which is extremely limited in nature. You have few milliliters or few millimeters cubes of tissue available from patient, which gets consumed or depleted every single time you run an assay. Besides this, the accessibility of these assays is further constrained by need for very high-end infrastructure, very specialist expertise, which leads to very long turnaround times and high costs, which make a huge impact when considering clinical implementation. As a result, more than 90% of drugs fail during development itself, leading to hundreds of billions of rupees lost every year for pharma, and more than 60 million lives being lost every year due to inefficacy of drugs in a clinical setting. So to address these issues, we've developed a very simple software platform that can take in routinely used bright field pictures of cells and tissues, which are ubiquitous in research in pharma and in clinic, including your H&E stained images. We analyze them using an AI-driven software to provide complete biological information, so your DNA, RNA, protein, metabolite changes, entirely digitally. So you get results at the click of a button in minutes. We overcome the challenges around limited tissue availability because we can create in silico multi-omics data. And this is a much more cost-effective way of understanding biology because it's a digital assay, not a physical one, and does not need very high-end infrastructure or expensive consumables. The underlying technology uses large-scale foundation models that have been trained on population-scale data sets, and they incorporate proprietary know-how from my research at King's College London and University of Cambridge on how biology results in different shapes of cancer cells and tissues. What we end up having is a software platform that can then be fine-tuned for a range of applications, all the way from research in academia to treatment decision-making in a clinical setting. And over the next few slides, I'll show you exactly how we can translate these or apply such a platform in these different areas. What we have today, for the very first time, is an opportunity where we can identify multi-omics or DNA, RNA, protein, metabolite information using a single technology stack without needing to switch in between technologies, equipments, or reagents. The underlying technology has several patents filed in multiple jurisdictions, with our first US patent already granted. And we've shown that this platform works on more than 30 solid cancers in a publication that was top 25 papers for 2024 for Nature Comms Medicine. We are using now the platform to really change how we understand disease by providing comprehensive multi-omics information across 5 different types of omics, and I'll show you a case study around that. We can leverage the platform for patient screening, whether that's patient sample screening in research setting, whether that's for drug development, so clinical trials, or for treatment decision-making, and we have more than 15 known biomarkers, well-established treatment biomarkers of cancers that we're already productizing. And we've also brought forward 2 products that have regulatory approvals in the UK market, one for breast cancer ER, PR, HER2 biomarker stratification, and the second one for colorectal cancer mismatch repair deficiency. Both of these have been validated in a real-world clinical setting and results published in peer-reviewed publications and are deployed in the clinic. So let me start off with showcasing how you can use some of these clinical products or biomarker profiling solutions in patient screening or treatment decision-making. The case study I'm presenting is that of our colorectal cancer product, which does mismatch repair instability testing entirely from routine H&E stained images. So you don't need any immunoassays or next-generation sequencing testing to be done. The solution is magnification agnostic, so you don't need any specific magnifications. It works on low magnification images as well. And the product is UKCA marked, so approved for clinical use in the UK, is currently available as a research use only tool outside the UK, and we are working with the regulators, CDSCO in India, to bring this to Indian markets. In a large-scale study across England, Scotland, Wales, and Ireland, we evaluated our pan-profiler colorectal solution, or PPC, on more than 4,700 patient samples, and it demonstrated a 94% sensitivity and a 92% specificity, which is very comparable to what we see with the wet lab tests, so your immunohistochemistry-based MMR assays or your next-generation sequencing-based MSI tests. Moreover, our solution was able to return results on an average of 80% of cases, demonstrating that it's broadly generalizable and robust to variations in tissue preparation, slide staining, scanning, and other processes that happen in the lab. So what you end up having is results at the click of a button in seconds, increased reproducibility, and it's globally accessible. The second case study focuses on understanding biology and how we can really develop better, more robust assays, but also understand biology when thinking about treating patients with the right sort of drugs or when developing drugs. In this case study, we took breast cancer samples from the TCGA. We had 992 samples. We trained the models to predict HER2 receptor status entirely digitally from H&E images. And then we assessed the agreement of our ML model-based prediction for HER2 versus the ground truths that was established by immunohistochemistry of FISH in cases of IHC2+ samples. What we found, as you can see at the bottom, is 119 samples that were false negative. That is, the AI classified those patients as HER2 positive, whilst the ground truth said those patients were negative. And we had about 30 samples which were false negative, so AI classified those patients as negative, whilst the HER2 immunoassays or FISH said that the patient was positive. And when we did further analysis, what we then started looking at was downstream effectors of HER2, so signaling pathways that are typically activated by HER2 signaling, which includes PTEN, AKT, and mTOR. And what we found was in the false negative cases, 71 cases out of 119 cases actually had downstream activated signaling pathways indicating active HER2 biology. So these were samples that were actually missed out and classified typically as negative, which were positive for HER2 signaling. Conversely, 20 out of the 30 false negative samples were actually had inactive HER2 signaling biology as indicated by inactive downstream signaling of HER2 effector proteins. And we've measured this both at RNA and protein levels. Collectively, we had 149 samples that were discordant with ground truth, of which 67% had either active or inactive biology collectively and were misclassified for treatment decision making. That means these patients could actually, there were patients who could benefit from treatment who were being missed. Majority of them, especially in the 71 out of 119 cases, and then there were patients who were not benefiting from treatment, who were actually given treatments increasing their risk of additional complications, including cardiovascular issues. So our solutions are further delivered as image format agnostic, so we support a wide variety of image formats. Doesn't matter what scanner or camera you use to generate the images. We deliver them as integrated solutions into existing workflow software. So if you have specific workflow softwares which you use to view and handle the images, we can integrate as a backend tool into the same. And the company is ISO 13485 and 27001 certified, meaning that we develop our solutions in alignment with international guidelines and standards ready for clinical care. We've already deployed our clinical solutions within the NHS, and whilst we have started making progress outside the UK, one thing I'd like to highlight is we've also signed a deal with one of India's largest path lab chain in bringing our clinical solutions to the Indian markets. Besides this, we also have growing relationships within the US with cancer centers like MD Anderson, and we work very closely with regulators, including FDA and MHRA, where we've actually worked with them and guided them in terms of establishing the right sort of regulations which allow for adoption of AI in an accelerated manner, but whilst ensuring patient safety. So this is ongoing work, and we are hoping to do similar work with the CDSCO as well. Besides this, we also have a growing pipeline of strategic data partnerships where we are partnering with large research organizations who want to use our platform for research use to fill in the missing gaps in the multi-omics data sets. And that's the sort of partnerships we'd love to bring here, especially in Gujarat today, which is my home state. And we have a very strong team with very strong expertise in the space, and we are backed by leading clinical and industry key opinion leaders, including Jakob Kather, who's a well-known oncologist and a professor of machine learning. He's a very big key opinion leader who's established the whole field. And Carl Barrett, who was former VP of Translational Oncology at AstraZeneca. So working very closely with both academics as well as industry organizations has been a key part to our growth. Before I close out, in alignment with what Dr. Ashok said, we would love to partner and bring and develop such technologies in India. So if you care about understanding disease better, or if you're working on developing better drugs or treating patients in the clinic, come talk to us. I'd love to understand and discuss how we can partner and accelerate forward. Thank you.
Thank you so much, ma'am. Your address has highlighted the transformative role of artificial intelligence in cancer diagnostics. We sincerely appreciate your time. I'll now request Ms. Pankti Mistry from GSBTM to present a memento to Dr. Bahini Pandya. A big round of applause, ladies and gentlemen. Thank you so much. Moving ahead, may I invite Dr. Somesh Chandra, Principal Medical Advisor, Clinical Innovation and Medical Affairs at IOTA Diagnostic Private Limited, to share his knowledge on the topic, Testing New Paradigms, Personalized Cervical Cancer Screening. Dr. Somesh Chandra is a veteran surgical oncologist and the Principal Medical Advisor for Clinical Innovation at IOTA Diagnostic Private Limited. With over 3 decades of clinical experience in complex robotic and cancer surgeries, he now lends his medical expertise to guide the development of innovative cancer screening technologies. May I request Dr. Somesh Chandra to join us on stage? Come on, everyone, can we please give him a big hand?
Good afternoon, everyone. I was listening quite intently to the esoteric scientific developments in the previous 2 lectures, and I'm mighty impressed by that. What I have to present to you is more simplistic. I am accustomed to working behind a mask in enclosed spaces, and in the company of such incisive minds, I get a little unnerved. So if the topic doesn't interest you too much, pardon me for that. But we have taken up this in earnest, and we'll tell you why it is to be done so. How do we change that? Pointing where? Okay. So these figures would tell you that there is a real need to address this problem, because this is persistent, and this can be prevented by following a simple dictum that to prevent cancer deaths, the best approach is to prevent the cancer. Working on such simple concepts, this publication in JAMA last year looked at the past 3 decades of cancer development and research, and found that while the proportions varied among different cancers. For cervical cancer, it was screening and prevention which contributed almost entirely to the reduction in cancer deaths. In keeping with that view, IARC and WHO have this plan on 90-70-90. Now vaccination is catching up, but for vaccination to really show a decline in cervical cancer mortality, we'll have to wait 50 years. What do we do for those 50 years? The first 10 years, obviously, we have to treat those who already have the disease. But for the next 3 or 4 decades, what's still going to work best is screening and prevention. So, where do we stand? Now, these are actually very shameful figures. The best is less than 10%. And to really make an impact upon cervical cancer mortality, we need at least 70% of screening uptake. And there are lots of studies, and this is an ICMR study published very recently, which enumerates what are the roadblocks towards achieving that screening uptake. So we are not talking about doing different things. We have to find out different ways of doing the same thing, which has been proven. Cervical cancer screening can prevent cervical cancer deaths by up to 90%. So basically, we need to shift our stance. And some people have been trying to do that, and you'll find that this was reproducible across all geographical entities. So the main issue has been the reluctance of women to go to a public health center to get themselves checked. But then we are putting the entire onus on women to get this done. There are a lot of other issues. There are issues about objectivity. A pap smear is a very subjective assessment. There are issues about logistics. There are issues about economics, about setting up such centers and training a lot of staff. So we need to bridge that gap. Some work has been done on this, and now self-sampling is now approved by the US FDA. And it seems that women could prefer to do it themselves, but then they have their own apprehensions about getting a proper sample or hurting themselves while they obtain the sample. And for this reason, it seems that women would rather, or anyone would rather have a non-interventional technique to collect that sample. So it's quite simple. So if we summarize all the roadblocks towards the cervical cancer screening uptake, we need to be more objective, which has been done by introducing HPV DNA testing as the preferred screening technique over the conventional pap smear technique, which is still being followed at many centers all over India. Self-sampling, better non-interventional passive sampling, a cost-effective, and a technique which takes care of lots of logistics issue, like transferring, storing a liquid sample. What the current technique is about a liquid-based cytology, and the HPV DNA is done through that. So there are a lot of issues with that. And here comes the second shift, and this is about doing it on dried blood. So that takes care of many of the logistic issues. The sample can be stored at normal room temperature. You don't need any special refrigeration. And it's very easy to transport it, and it can be stored for long periods of time. Is it suitable for HR HPV testing? Apparently it is. There are a fair number of convincing studies that dried blood sample is a good sample for screening as well as genotyping of diagnosed cases. And this is a review published by the Tata Memorial Group, 5 studies quoted. One is a very Indian study, a large study, population-based, 500 people, subjects in that. But these tests, these techniques are not standardized, so they are not reproducible. And we cannot put a population at large to subject them to a method which is not standardized and reproducible. So we needed to make a working model to make a reproducible cervical cancer screening test using dried blood. And this is where 3 of us came together, leading lab in Ahmedabad, Weber Shitole from IOTA, and we sat over and devised this. IOTA already had biosampler, and it needed to be modified. We call it M-strip because it's stuck on the menstrual pad, and it absorbs the menstrual blood, and from that the HR-HPV testing is done. It is a CDSO approved tool, and it is now patented in India. So this we needed to know. We knew that dried blood is a good sample. We need not rediscover the wheel. What we wanted to know was whether this was a feasible and acceptable to women. So we did a feasibility and acceptability study, and we found that this promises to improve drastically the uptake of screening. And while efficacy was not the prime objective of this study, we found that there is 100% concordance with positive and negative cases. So these are some of the sample findings that, yes, women here in Gujarat, this was done, would also prefer self-sampling. And there is a Stanford study using what is called a Q-pad, where the entire sanitary pad is provided. So we specifically asked women whether they would like to have the whole pad or a strip, and clearly the strip is the preferred method, and because most of the women would prefer to use a sanitary pad of their own choice. So we published it this year, and in less than 6 months, it is one of the most viewed articles. So apparently it is catching interest, but this is a new paradigm. Women were normally subjected to a cervical screening in the intermenstrual period. We are doing it during the menstruation. We are using dried blood for the first time. This sample comes directly through the cervical canal, unlike self-sampling methods currently in use, which are primarily using vaginal sampling techniques. This is non-interventional, and this is now a very standardized technique. We have done one more study on this, registered with CTRI, and the concordance is very much in line with our previous findings. This opens up new horizons for us. The Australian cervical screening program has already embarked upon a risk stratified program. So from testing all women at risk, all women who are sexually active and beyond 21 years of age, up to 65 years of age or beyond, you can bring that number down to 10% who are HR-HPV positive. From the HR-HPV positive, you can look at those who are HPV 16 and 18 positive. Subject that small number, which would be 2% of the entire cohort, to a more intensified screening, because these are the subtypes which are more likely to be associated with CIN2 or a higher stage of changes. So, and the rest HR-HPV can be monitored at more frequent intervals, can undergo pap smear and cytology. So this is a risk stratified based screening that we propose. And it gives women that liberty, that freedom, and that convenience to get away from a blanket approach and be more proactive about their health. And these are the limitations. So this is obviously for menstruating women. But if we remember that the peak age of incidence of cervical cancer in India is just beyond 50, that's the 6th decade. And it takes about 10 to 20 years for the disease to develop. If we focus on the premenopausal group, which are menstruating, we would catch them at the right time for disease prevention and appropriate treatment. Post-hysterectomy, the risk of vaginal intraepithelial neoplasia and vaginal cancer is very low. Actually, hysterectomy reduces the risk to about hardly less than 5%. So they are not very serious concerns. We found that social taboos, so while we have got the tool, it's still there in the mind of majority of women who refuse to undergo screening. These are the issues, the social taboos and the beliefs about menstrual blood, which could come in the way. There are other roadblocks we found during the study, that communication and repeated reminders are very important. Even because you have a window of, say, 3 days in a month for women to provide a sample, and they could be busy, they may miss that cycle. So we need to devise those reminder programs also, and it's better incorporated as a comprehensive program. So what we propose now with partners who are active in community health, who are doing social service, and possibly some modern diagnostic tools like AI-based colposcopy is what we call the C-TIPS. TIPS is our program of targeted intensified program for screening of the commonest cancers. That's cervical, breast in women, oral in men. And maybe on a different time in a different forum we can talk more about that. So to get, when we put small acts together, we get a very different picture. Thank you very much for your patient listening. If there is time and if there are questions, I'm ready to take them.
Thank you so much, sir. We are truly honored to have a pulse of wisdom from a veteran personality as you. I'll now request Dr. Kunjan Kumar Mehta from GSBTM to present a memento to Dr. Somesh Chandra. Can we have a huge round of applause, ladies and gentlemen? Thank you so much. Before I announce for the next speaker, I would like to request the speakers of the day. We all need to stick to the timing because we have so many presentations, and then at the end of the day we have the panel discussion as well. So I request all the speakers, please stick to the timing. May I have a privilege to invite Dr. Pawan Gupta, Director, Surgical Oncology, Max Institute of Cancer Care, to address the audience on the topic, Translational Progress in Head and Neck Cancer. Dr. Pawan Gupta is the Director of Surgical Oncology at Max Institute of Cancer Care, an accomplished head and neck oncosurgeon with over 25 years of experience. He is also recognized for his social activism and innovations in cancer awareness and tobacco cessation campaigns. May I request Dr. Pawan Gupta to take the stage? Welcome, sir. Round of applause, ladies and gentlemen.
Thank you so much, and I first want to thank Dr. Anshuya for inviting me for this talk. And speaking after Dr. Shomesh, it's really, I'm getting unnerved now. Thank you. So we are going to talk about, first let me tell you, I'm a clinician. I'm a total clinician. So many of you are researchers. What I'm talking about is going the other way around. I have been visiting a lot of research centers, and what I have basically done is asking them to give some solutions to my problem, right? So what I will be talking about is basically a assimilation of things that I have generated, getting means collaboration with a lot of important centers across India. So this is basically, I'm talking about head and neck cancer, but mostly focusing on oral cancer, right? Oral cancer, because I call this as the king of all cancers in India. We are the highest in the world in oral cancer. And wherever we go to any centers, we are still, if you talk about lung cancers, you talk about breast cancers, you talk about colon cancers, so many other cancers, basically we are taking all the important aspects from the Western world and we are incorporating into India. But when it comes to oral cancer, we are supposed to be the leader, and we need to tell to the West what they need to do. The number of cancers that we see in India, probably they may not even see what we see in 1 month, they will not even see in 1 year or even multiple years. So this is what I am quickly going to say, and I'll try to stick to my time only. So please tell me before 5 minutes before so that I can fashion up, right? So this is, I am also representing the International Photodynamic Association, right? I am the ambassador for India for the Photodynamic Association. And like the mission, Gujarat State Biotechnology Mission, so this is a mission that we have actually started, that is the mission stage 0, right? Right now, what we see, oral cancers mostly coming at the stage 3 or 4 to us. And we have been talking about early screening, early diagnosis. So oral cancers is one where we have started this mission stage 0, and we are going to launch it in maybe in February. This is just, we are developing the whole mission, how to go about it. And it's a war against oral cancer, right? We have to see, and we have to win about this war. And so there are 4 components about this, on this war against oral cancer. The first one is the tobacco marshal, right? Everyone becomes a tobacco marshal in the society, so we have developed an entire training program. It's just not about volunteering, but it is about understanding about tobacco and how to help people or how to guide people to quit tobacco. The Oral Oncology Connect program is a capacity building program where we get most of the doctors, that's the Oral Oncology Connect, dentals and the oncologists, where they connect on a platform. We have developed the entire platform, and we train them so that they can diagnose these cancers at the early stage. And Reach Out for Zero is a health screening, that is a Reach Out for Zero, that is screening program that we are conducting at various places in community. And what I'm going to talk about is the innovative treatment, that is the photomedicine. Right, this is what we are trying to. So what, actually, when we do the screening, I have been talking to a lot of people in the morning, and I have been talking to multiple places. Most of the time, we end up with screening. But the entire cycle has to be completed. Just screening is not enough. We have to treat these patients after screening. If you pick up these lesions, we should be able to treat this patient and see to it that they do not get cancer in the future. If we have not done that, then it is incomplete. We are just giving them a screening and telling them कि देख तेरे को cancer हो सकता है, but उसके beyond हम कुछ नहीं कर रहे, right? So this is where we have to, as a clinician, this is what we are looking at, how to develop the entire program till the end. a full cycle, full circle. So this is just, since I'm going to talk about photomedicine, these are some of the pictures. You can see this. This was the lesion, right? This was the lesion early, and we have treated with photomedicine, FOSCAN. Dr. Shomesh was one of the pioneers in FOSCAN treatment. So after this treatment, this patient is completely cured. This patient was otherwise supposed to have a total glossectomy. That means the entire tongue was supposed to be removed in this patient. But with this treatment, this patient is alive and doing his work completely. This is another area. There is inflammation in the oral cavity, which can be erythroplakia, so many other lesions which can be tobacco related, non-tobacco related, and this can be treated with photobiomodulation, that is PBM. And this is what we have shown with PBM. There is a complete disappearance of the lesion. And this is one simple technique that we are using. The disease kept on increasing, but with simple tobacco quit program that we have developed, the patient was completely healed and he's doing well and he's completely off. So when we are talking about photosciences, it's a complete picture that we are working at. It's working at the diagnostics through, showing that autofluorescence, spectroscopy, that is the optical biopsy that we are talking about. The therapeutic, that is photobiomodulation, photodynamic therapy. The supportive, that is for the radiation or the medical therapy that is given. So for that, the mucositis can be treated, and of course the preventive, that is the tobacco cessation, and potentially malignant lesions can be treated with this methodology. So this is just, because I'll have to go quickly, so this is just a diagnostic spectroscope that has been developed with the aid of IIT Delhi, and they are showing that how these pictures can be picked up, and with the AI based, we can diagnose as an optical biopsy whether it is pre-malignant, whether it is malignant, or it is just inflammation. And this is a photobiomodulation. Again, I'll not go into the details, but this actually helps to heal up. The light therapy, a specific laser wavelength therapy is given where the mucosa heals up faster with the ATP production. And this is the photodynamic therapy where three things are important. That is the photosensitizer drugs, oxygen, and the light. Right? With that therapy, the specific cancer cells are killed. Right? So these are the light therapies, which are again various. It can be laser, it can be lamp, it can be LED. It has got all these plus and minus points, but I'll just quickly go through. And these are the photosensitizers that are required for the photodynamic therapy. That is, if they are pre-malignant lesions, we can treat these patients. We just do not have to observe them, we need to treat them. So PBM applications are various, basically for inflammation. PDT, the application is to kill the tumor cells. So this is the model that we have developed. High-risk screening, clinical oral examination, we can use the photodynamic detection, that is the diagnostics, which can autofluorescence, white or blue light, spectroscopy, divide the cells or divide the patient, group them whether they are normal, inflammatory, high-risk, potentially malignant lesions, or malignant. If they are malignant, they go to the cancer treatment, proper treatment, whatever it is. But if it is high-risk malignant, they can go for a early photodynamic, that is a topical photodynamic therapy. And those who are inflammatory, they go for photobiomodulation, and normal will go for follow-up. So this program was developed in association through USISTF award that we got in 2024, and this has been developed in association with US centers. I'll be talking about that. So this was one of the cameras that was sent from US to us from the University of Arizona. But this spectroscopy was developed indigenously at IIT Delhi, and we are now collaborating with them. It is already, they have applied for patent, so probably we will be incorporating into our system. And this was again a developed methodology on LED-based, very cheap method. Hello? Okay, for treatment with photodynamic therapy of these lesions, and the trial was done at Aligarh already. Now we are adopting it, and we are looking at CDSCO. So this is again another important this tobacco cessation program, which I had worked quite extensively along with, of course, Dr. Rakesh, and we have been doing this research for quite a long time. And what we have developed is a complete systematic program by which we are able to treat these patients in a holistic way so that they are able to quit tobacco over a period of 3 months. And this was what we wanted to develop, an integrated technology system, and that is what we are developing in India, so that it can be put in the rural centers, right? Because oral cancers is mostly seen more in the rural centers, so this cheap or the inexpensive device that we are trying to develop in India itself, indigenously, and we have already almost developed it, so we want to put it in the rural centers for the patients. And this was presented in Shanghai in the IPA 2025. Actually, with Jonathan and others, I met in one of the IPA in Finland, where we started working that how we can work in collaboration to develop this device in India itself. And this is, I visited personally to this laser manufacturing companies in China, and it was found that just 5 minutes, okay. So this is where we have started working with them, and then we visited number of hospitals in China itself where this technology is being applied on the patients, and also this being applied in the European countries as well as in US countries. Just 5 minutes, I'll just. Okay, so these are the research papers we have developed. So what we are looking at, integrated development of one single system for oral cancers, and it's not just diagnostics, just screening, but complete therapeutic, right? So it completes a 360-degree from prevention to complete therapeutic, that is what we are looking at, and it's basically, it should be standardized, scalable, sustainable, and sellable, right? And that's what we are looking at. So this is some of the collaboration that we have. I'll just quickly go through, and this is the biosensor we are developing with IIT Jammu, right? And this is a biosensor which is for quantitative and qualitative estimation of nicotine in saliva. With just a biosensor, nicotine in saliva, so that with that measures, we will be able to intervene the type or the medication that is required for them to quit tobacco. And they can be followed up whether they are actually off tobacco or not, whether they are telling lies or truth, that also we can find out, right? So this is again laser and photobiomodulation for oral lesions related to tobacco. This is what we are actually working on. Use of laser for tobacco cessation. That is another photobiomodulation for tobacco cessation, because people don't want just assurance, they want some treatment. So this is another treatment which is, we have already tried on some of our patients, and it is working wonderfully. It has been tried in Western world, but we are trying it in India, and it is working wonderfully. Use of oral probiotics for tobacco lesions. I think Nirma people and others, we can work with them on that. The mechanical mouth opening device for submucous fiber, it is already patented and in the market. And optical biopsy for oral potential malignant lesions. This is something. And the another important is development of the so these are okay, quickly. So they went. So these are some of the pictures that is with the IIT Jammu, where we are developing the biosensor. This is with the Santosh University. What we are doing is the screening program in the community. We have had an MOU with them. And with the MedSol company, which is from Hosur, we are actually developing the laser products indigenously. What is costing almost like 20 lakhs out from outside, when we are making it just for 1 lakh in India. Right? So that's them. And this is the device which is patented, thanks to Dr. Rakesh. Actually, we developed this in Gujarat, JUSEC. And now this is a patented product. It's a very simple device, mechanical device, but it is now patented, right? And of course, this is the one device which is also now filed a patent, and it is for the oral screening, that is the spectroscopy. And of course, this is again cessation app, which we have developed. And we have shown that the cessation pattern is almost 46% in 2 years. This is important. And what we have now started is a single helpline number, that is 977-385-6664. Any person, wherever we are going, you can just put your picture on this WhatsApp, and our group of doctors will be able to respond at this number, what is the lesion, right? So we are going to automate it very soon with the app, but right now we are testing it on our WhatsApp group. So just to end, oral cancers has a long pre-malignant stage, screening and early detection, of course. 50% of PML reversible by tobacco cessation itself. So tobacco cessation has to be incorporated in any screening program that we are working on oral cancers. Right? And tobacco cessation and PBM, that is very important that we can do for the lesions. Treatment of the residual, that is with laser electrocautery or cold blade excision, needs long follow-up. This is what we are actually focusing on, that these patients, they need to be followed up on a long period of time. So each patient, the photograph is taken of this patient, the details are taken, and we are following up this patient on a regular basis for at least for 5 years, 10 years down the line. Right now we have done for 2 years for a lot of our patients. So this entire app has got these pictures, and this, what is the lesion? Typical 8 pictures are taken for each patient, at each encounter. So for the late stages, of course, after the surgery, radiotherapy and chemotherapy, we have to tobacco cessation is must. Patient has to be followed up again, and if there is a recurrence, which is very common, it can be treated with PDT, photodynamic therapy. So surgical intervention. So it's a dream to reality. That is for, we are looking for more partners. Today morning we were talking to the Nirma group in the morning, so that the one very important thing that we are looking at, the immediate challenges that we are facing is the ALA. If we are getting ALA, that is a photosensitizer, it is very costly. Either we have to get it from China, Germany or Ireland, and it is very costly. It may not be affordable for our patients. So we are, and what I understood from Rakesh ji, again, Dr. Rakesh is our, every time he keeps helping us. So ALA is a very simple molecule, which can be developed easily in India. Either it can be synthesized chemically or it can be biosynthesized, as he has said. So we are looking for partners who can help us make this ALA in India itself. Right? Maybe the Nirma group or any other, we can talk to you. So this is one thing that we are taking. Of course, the regulatory approval is another important point. The other things that the local manufacturing, others, I think that will work up. But two important, someone who can manufacture ALA in India. The other is basically the regulatory aspect, which I think Purnima ma'am, we can talk to you. Right, so that's it. Thank you so much, and thank you so much.
Thank you so much, sir, for the comprehensive and thought-provoking address. I'll now request Dr. Daksha Sakhia from GSPTM to present a memento to Dr. Pavan Gupta. Thank you once again, sir. Thank you so much. Can we have a huge round of applause for him? Thank you. May I now request Dr. Mayank Singh, AIMS Delhi, to address the audience on the topic, Developing Adaptive Cellular Therapies in Public Sector Institute, Opportunities and Challenges. Dr. Mayank Singh is an additional professor in the Department of Medical Oncology at AIMS New Delhi. With extensive postdoctoral experience from leading US institutions, his lab focuses on cancer immunotherapy and understanding the tumor microenvironment to develop novel therapeutic strategies. May I now request once again Dr. Mayank Singh to take the stage. Can we have a round of applause for him, ladies and gentlemen?
Good afternoon, everyone. So I will try to stick to the time limit before I guess I'm the last speaker before the lunch begins. So I will try to stick to a time limit as much as I can. So yeah. So today I'm going to talk about this work which is developing adaptive cellular therapy in public sector institutes. So I think from the morning we have been talking about different forms of cancer, like the way initially Dr. Ashok started and Dr. Amit touched upon his work on the lung cancer, and now we were hearing the surgical oncologist perspective. So different varied field regarding the cancer biology and how it works. So our work basically we are working on the adaptive cellular therapies in cancer. And when you talk about adaptive cellular therapies, what we mean exactly is that we mean the cellular therapies. But if you see, it covers a different field, right? Once you talk about the therapies, we hear about the cell therapies, we hear about the gene therapies. So, but there is a difference between cellular therapy and gene therapy. Cell therapy is something when you take out a cell from a human body and you can actually modify it and you put it back into the human body, thereby they can actually target a particular, the target of interest, which is the case with the CAR T cell therapies, which I'm going to discuss about today. Then you've got the gene therapies, which basically refers to the removal of anything replaced by genetic material in the human body. So those are the key differences I just highlight here. So this is like transfer of the whole functional living cells. So CAR T cell therapy comes under the realm of what we call as the adoptive cellular therapies. Oops, sorry. So if you see what actually the CAR T cell therapy is, when we talk about cancers, and like about 20 years back when somebody used to talk about cancer, it used to be sort of like a death sentence for a person, that whether it's going to get cured, it's not going to get cured. And what we used to get is that you used to have the therapies which were basically non-specific, and that is what used to happen is that when you treat the cells with any form of say what we call as chemotherapy or a radiation therapy, what happens is that it is causing the apoptosis of the normal cells as well. So that is why you used to get a lot of toxicities in the patients. But it has changed a lot now. Today what has happened is that 10 years back there was the advent of the immunotherapies. What exactly are immunotherapies? What immunotherapy is going to do is that it's going to go and actually target a specific cancer cell, and without causing a toxicity to the normal cells of the body. So that is why you get like your low toxicity values. Now the immunotherapy field has also progressed ahead. So now what we call is that adaptive cellular therapies which are present. And in the adaptive cellular therapy, what you do is that, and the CAR T cell therapy, which we call chimeric antigen receptor T cell therapy, is one form of that therapy, which basically targets a particular antigen on a cancer cell. If you see, this is how it looks like. You have your T cell which are isolated from the patients. These cells are isolated, and after that, these T cells are actually modified outside the patient body. And then after that, you infuse these cells back into the patients, and then you observe you expand these cells, you train the cells to recognize their antigens, and then you put them back into the human body, so where they can go and actually kill the tumors, and you get rapid form of a tumor apoptosis in these cells. So if you see the mechanism of the CAR T cell, this is how it looks like. These basically water cars, basically. That's fine? Yeah. Okay, that's okay. Thank you. I think this gives much more freedom. So when you see these CARs are basically genetic engineered fusion proteins. They have antigen binding domains which are derived from a monoclonal antibody. And then you have an intracellular T cell co-stimulator domain. So what we are basically doing here is that we are taking this antigen binding domain from a body and fusing it with an intracellular T signaling domain. So this is classical immunology what you are doing here. And this is how it looks like. Once you have these ones expressed on the T cells, you give them to the patients, they go and they destroy the tumor by the classical immune pathways. But the CARs have respective advantages and disadvantages. Is that one of the main disadvantage of CAR is that they can actually only recognize the cell surface proteins. So we don't have any CAR T cell therapy again intracellular antigens up till now, but we have for the surface antigens. One of the main advantages is that they don't require antigen processing, so they are HLA independent, so you don't have to do that. And these are more secure, better targeted, and more persistent. What you see here, that the concept of the CAR is not new. It's a pretty old concept, and it was there for a very long time. In fact, the Ishar et al. Back in like 1980s, they showed that actually you can synthesize this fusion protein, and you can put on the T cells and you can infuse them into the patient. But what happened was that these T cells were not persistent. They died out. They were unable to do the tumor toxicity. So that concept died at the time. And after that, what happened is that there was an advent of what we call a second generation CAR. So what they did in the second generation CAR is that they introduced a second co-stimulatory domain. And these are the CAR which you see in the market today. All the approved CAR T cell therapy which you see in the market today are second generation CAR T cell, whether it is CD19 or whether it is BCMA targeted cell therapy, because these are like much more secure. Then we have the third generation CAR, but what happens in the third generation CAR is you have 3 co-stimulatory domain. But what is happening here is that you get a massive antigen stimulation. Because of that, you get a big CRS, and because of this big CRS, the patient actually collapses sometime. So these are different types of CAR which are basically available. If you see here, the first generation CAR, second generation CAR, third generation CAR, and these are already there. The majority of the FDA approved CARs which you see in the market are the second generation CAR. Then there's a development of the fourth and the fifth generation CAR also, which basically, these CARs are basically developed for the solid tumors, because as of now, we don't have any approved therapy against the solid tumors in the cancers. But these are basically what we call armored CARs. So they can actually, like these T cells actually can survive in the harsh tumor microenvironment. So if you see Y CARs, these are best of the world of immune systems, like these have B cell specificity. and then T cells hydroxy without presentation is a form of adoptive cell therapy. They are synthetic engineered receptors and they target surface molecules. So one of the most important aspects when you work with the CAR T cell is your selection of your antigen. The antigen should be very, very precise. Because see, when you develop a CAR, you are not actually developing a CAR therapy against a particular cancer. You are developing it against a particular antigen. If that antigen is present on a normal tissue, then the CAR T cell is going to destroy it. And that is why you get a lot of toxicity in those. So ideal CAR T cell should be tumor specific. It should be universally expressed only on a tumor cells. It should be a cell surface molecule. And two of the molecules which have emerged in recent time is one is CD19, against which you have approved like 4 different products. Then you have a BCMA, which is called B cell maturation antigen. It's there. And the third one which has been developed is HER2, which has been developed in different form of cancers. And this is how it looks like. If you see the CAR targeted hematological cancer, this is how it looks like. If you see BLL, TLL, AML, or CLL, you are seeing CD19 is there. But if you see in multiple myeloma, the BCMA expression is pretty high. So that is why the approved product in the market are against the BCMA. And these, if you see, this is the list of the FDA-approved CAR T cell therapy as in the market. If you see, there are now 5 different products, CD19 targeted CAR T cell available in the market with varied kind of antigen specificity, toxicity profile, and that. If you see against the BCMA, which is called a B cell maturation antigen, you have got 2 different products which are available in the market. So our work basically covers the development of the CAR T cell in multiple myeloma. If you see briefly what multiple myeloma is about, this is how it looks like. It starts with your MGUS, which is mononuclear gammopathy of unknown significance, and after that it produces all the way to plasma cell leukemia. But what is happening during this time? There are genetic changes that are happening in the cells, and because of the genetic changes, what is happening is that the expression of the surface molecule is changing. And one of the expression which changes with this is of BCMA. And this is how it looks like. This is the first line of therapy. When you give this patient the first line of therapy, there is an emergence of the resistance clones in this. When you give this patient the second line of therapy, there is an emergence of further resistant clones. And then after that, what happens is that the disease becomes what we call a relapsed and a refractory disease. So there's a relapse, and it won't respond to any target whatsoever you use in this case. So that is where the CAR T cell therapy comes into play. When at this point the patient will have limited options, you can give them the CAR T cell therapy, and it's going to work in that. And one of the molecule expression changes as the multiple myeloma progresses is BCMA, which is B cell maturation antigen, and the higher level of a BCMA are associated with poor outcome. It has a restricted expression on the plasma cell, and currently there are multiple emerging BCMA-based strategies which are being approved in the multiple myeloma, and this is how it looks like. If you see, these are different BCMA-based immunotherapies, there are BCMA CAR-Ts, ADCs. You talk about NKs, BiTEs, and different form of that. And this is how it looks like. This is a list of the different clinical trials which are basically targeting the BCMA in the form of ADCs or the CAR T cell therapies and different whatnot. And this was the trial which changed everything. The snapshot which I'm showing you is from the KARMA trial, which was done after which the CAR T cell were approved in BCMA. And this was done in the relapsed and refractory BCMA patient. If you see, there was 72% overall response in these patients. So thereby these therapies are very, very effective in this patient. After that, the KARMA trials, new trials, KARMA 1, KARMA 2 has happened, which have actually enrolled different set of patients and giving very, very promising results. So we started our work at AIIMS back in 2021. So we thought, okay, we need to develop a CAR T against BCMA. The first question which we asked ourselves was that, how do we start it? So the first important thing was to develop an antibody, obviously. We first need to develop anti-BCMA antibody, out of which we are going to synthesize your binding domain. So we thought we use 3 different approaches. We will use hybridoma or a fast display, or we will just source out from some other source. But finally, what we thought, if we want to patent this thing, so we should stick to the classical approach, which is the hybridoma technology. So we use that approach. We developed the antibody, and this is how our CAR looked like. What you're seeing here is a structure of a CAR cassette which we use. If you see, this is a second generation CAR. As you can see, there are 2 different domains, 4-1BB, you see the 3 zeta. And this is, in the middle, what you see is the anti-BCMA CFE, which has been derived from an antibody. So this is how our CAR structure looked like. And after that, we did that. So finally, the clone which we got after repeated screening, and screening was a 9C4 antibody, which was out of the best binder among the 20 clones which we generated at AIIMS. After that, we took the antibody out of this 9C4 antibody. We took their SCFE, which is small gel variable fragment. We patented this antibody, and we actually conjugated with a T cell receptor to generate our chimera, which is called a CAR. So this was basically the 9C4 CAR. And this was obviously this was second generation CAR, so we cloned it into the lentiviral vector, which is a P lentivector. We encapsulated this CAR into lentiviral particles, and we took the T cells from the patients. And then we do it. So this is the whole workflow which we have generated through that. So if you see, it has got first antibody generation, then the sequencing of SCFPs, then cloning into the lentiviral vectors, subsequently production of the lentiviral vectors which have these CAR. And we took the T cells out of the patients who were coming at EAMS, expanded those T cells. We transfected these with the CAR to generate finally the CAR T cells, and we have actually, so we have completed this whole workflow, patent this CAR, and I think this, more or less, this thing is ready to go into the phase 1 clinical trials. That is what we believe. But during this whole process, what we learned out of these things, we learned that there are different challenges which are associated with doing these kind of things in the public sector institutes. And obviously, if you see the whole thing, the way I'm talking about is that the complexity of the manufacturing process of the CAR T cell. It is a very complex process. So for that, you require good GMP grade facilities. You require proper scale up. You require proper lentiviral vectors and all those things. So obviously, because the CAR T is individualized, your multiple complexes are needed. Automated and closed systems can be used, but they are not good for a longer period of time. You need like larger production facilities. If you see the cost and infrastructure, the high cost per patient, I'm talking about the US perspective here, it is like exceeding to 300,000 to 500,000 is a core challenge. So you have to look at different aspects, how the production will be done, how will you produce the GMP grade vectors, how will you produce the GMP grade CARs, how will you do the transfection, how will you expand them, multiple processes. Additional hidden costs are also there. When you work at an institute like AIMS, you have got different advantages also, and if you can take care of that. But the most expensive step in the CAR T production is obviously production of the viral vector. This takes like as much as over $100,000 per infusion. So that has to be addressed, that thing, and that is a tremendous bottleneck which we have seen actually. If you see the total costing distribution, this is how it looks like. FRS costs you up to $10,000. Viral production goes up to $100,000. Modifications, cell culture, quality control and testing, very important in this case because you need to give this product to the patients, right? Then cryopreservation. So these estimate highlight why cost reduction effort are focusing more intensely on the viral vectors. That is what we are also looking up. How do we scale up the things? How do we generate quality generation vectors so that we can take the patient? And obviously, if you see the lentiviral cost per USD, it is like, if you see that, like the transfection units, it's very high. So what are your sourcing options at this point of time? How you can source these things? So major suppliers include specialized contract manufacturers, CMOs, offering GMP grade lentiviral vectors. Industry leaders also provide GMP grade lentiviral vectors, like different companies they provided, but those are very expensive. You cannot rely them for a longer period of time. Manufacturing can be done in-house with bioprocessing capabilities, outsourced to CMOs. What we see here is that there should be a proper model for public sector, private sector in that. Then continuous development in the production method, including stable produced cell line and scalable transition aim to reduce cost and improve the yield. That is one way ahead. Then you have to do quality control and release testing, very important thing. When you have a product, you need to test it for different viral particles. You have to produce something which is called as a industry grade or a patient grade material, which requires a lot of testing. It takes up a lot of things. Then you have a vein-to-vein time, which is your centralized manufacturing and different product, cold chain logistics. Then you have got regulatory hurdles also. Like when you work in a public sector institute, often the question is asked is that you are not an industry, right? How will you ensure that everything is good here? So that is one challenge which we face day to day. So those are the things. But if you see the case of adoptive cell therapy, what are the key challenges in Indian scenario? The development of academia and industry consortia is one thing. The focal point should not only develop a CAR T cell, but also development of vectors. That is one thing which is very important. When you develop a CAR T cell, you need to work on the vectors. So how are you going to deliver the product? The clear-cut cost-sharing system need to be devised. The focus should be on the in-house production region so that reliance on any particular source can be avoided. I think that is very important for a long term, when especially you want to work in bigger institute like AIMS or any other place where you have tremendous patient load. That's not changing. Yeah. So this is how it look like. So these are the CAR-T challenges. If you see, this is the figure which I have taken from our journal. So these are the challenges, but these are a different way. Many times a question comes to me, why does the CAR-T cell therapy have not been developed for the solid tumors? So this is why it looks like. This is why they are not developed for the solid tumors, because the solid tumors have a hypoxic core, in which the T cell does not work that well exactly. You have got the challenges, how a T cell is going to go to that place. Then you do not have ideal marker in the solid tumors. So what happens is that there's a lot of expression of these on the normal tissues. That is why it cannot happen. So those are the things. So if you see about the B-cell CAR-T, what we have learned about the resistance mechanism in this case is that sometimes what happens is that you're developing a CAR-T against a particular target. What happens if the target is not there? And that is what happens in this case. You have a case of BCMA negative relapse, which happens in these patients. So we are trying to develop what we call as a bispecific CAR-T cell, which is funded by Department of Biotechnology. And this in which we are developing an anti-BCMA SLAMF7 CAR. We are also looking at the future, and we are seeing that we can have those answers whenever the things come in. We have those things ready. So then we have obviously the CAR-T cell bispecific antibodies. We are also working on the ADC drug conjugates to develop. We are using this antibody conjugated with auristatin and developing an ADCs. So this is our immediate and long-term effect of research group. We have generated a preclinical second-generation CAR, which we have filed for a patent. Now we are trying to take this project to a phase 1 clinical trial. Let's see how it goes, how much support we receive from the funding agencies for that. But this is an important question. If you see the ID cell, BCM-based CAR-T cell therapy, it cost in US is $419,500. That is how much it cost, which is very, very high. So we are working to bring this cost tremendously down. The other groups who are working in this field in India, they have brought a cost possibly like very low, but I think that needs to go down further so that these kind of, I think the breakthrough treatment are like available to the public of India. That is what I feel think. So thank you so much for your time, and these are the different people who help me out. We have got collaborators in Indian University in Houston, and obviously BBT funded our project, ICMR also funded the project, and we are working with different departmental teams, biochemistry. Then we have got the clinical collaborator Ranjit Sahu and Rajan. They're helping us out. Thank you so much for your time.
Thank you so much, sir, for your valuable address. Now I'll request Mr. Harbhadan Gadvi from GSPTM to present a memento to Dr. Mayank. Can we have a big round of applause, ladies and gentlemen? Thank you so much. All right, ladies and gentlemen, so now it's time for a much-deserved lunch break. So because we are running a bit late, I would appreciate if we all can reconvene in 45 minutes. So probably we are starting with the next session at 2:30 sharp. Thank you so much.
and see what really the numbers are. And the reason why we started working in India is because India has a really specific genetic architecture. We were, in fact, the very first group in the country to identify the founder mutation for Lynch syndrome. We just did that study late last year. We showed that this mutation emerged nearly 800 years ago. Nearly 6.4% of all Lynch syndrome patients, this is patients generally from North Indian origin, have this mutation. That equates to about 3 lakh out of 46 lakh people with Lynch syndrome in the country. And this really helps in developing targeted diagnostics. You really reduce the cost substantially when you come across it. And of course, we ended up on the Times of India newspaper for this. So, when it comes to pan cancer, we wanted to look at nearly 300 patients all across India with all different types of solid tumors. And we had a wonderful set of clinical collaborators that I can't thank enough. But what sort of MSI testing should we do to enrich Lynch syndrome patients? Well, here is the CAP guideline. It suggests that if you have colorectal cancer, you can just do MSI by PCR because that's the standard of care. But for remaining else, the jury is still out, whether you do IHC or whether you do PCR-based analysis. So we said, let's do both. So for every tumor that came through, we did both MSI by PCR and immunohistochemistry. If both tests showed that you are MMR proficient, no further testing was done. But if either of them were positive, we would do germline testing to identify Lynch syndrome. And here is preliminary data. The study is still ongoing. The red bars show percentage of patients who ended up with MSI high tumor, stratified by which tumor type they are on. The horizontal line indicates it's the benchmark for colorectal cancer, but clearly you can see that there is substantial enrichment in extracolonic tumors, especially endometrium, small intestine, and liver. Now, if I look at percentage of Lynch syndrome across the entire cohort, regardless of MSI status, it's about 6% of the overall cohort. But the moment I use MSI for enrichment, it's nearly 42% of MSI high patients who have this. It means MSI is an excellent biomarker to enrich these patients. Now, here is a tumor type in which we identified Lynch syndrome. And as you can see, majority of them were colorectal cancer patients. But here's a catch. Nearly 50% of them had a really interesting variant. It's MLH1C.154del. This variant kept on coming again and again and again. The interesting thing was that they were all from Gujarat. We didn't find this variant anywhere else. But if we exclude all of these patients, just like Caucasian population, nearly 50% of the patients had extracolonic tumors. It means we are missing a substantial number of patients if we just focus on colorectal cancer. So, C154del, really quirky stuff. So, my PhD student one day went back to the clinical notes, came running to my office and said, "Sir, there is a problem. All of these patients are seen by one doctor, and they all belong to one particular region of Gujarat. Their surnames are different, but they all belong to one particular part of North Gujarat." So he said, "Okay, let's do most common ancestor analysis." 2 more minutes, please. Thank you. And we identified that these patients were related to a common ancestor nearly 15 to 25 generations ago. So they were very, very closely related. When we did phasing and haplotype analysis, it turns out that they were all sharing one same haplotype of nearly 700 kilobase in size, and this originated nearly 70 generations ago. So we have second founder variant that is very, very specific to Gujarat, that also very specific part of Gujarat, that helps you to do targeted screening in those populations. Well, coming to extracolonic tumors, as I said, why do we look into that? Because you have the highest risk of death. So this is part of our large study with prospective Lynch syndrome database, and we show that if you were to just focus on colorectal cancer, you're missing out on a huge amount of mortality that occurs in upper GI cancers. So, coming back to CAP2, it was not one, but a 2x2 factorial trial. At the end of the day, I'm Gujarati. I love to save money. And because it's a 2x2 factorial trial, half of the patients were given resistant starch, equivalent to 2 slightly unripe bananas a day. The remaining were on placebo. They were then followed up for 20 years, and hey presto, at 20 years, there's a 50% reduction in upper GI cancers. So pronounced was it that we ended up on the cover with bananas. This is the journal cover. The first time it happened for us. Not only that, when we followed up these patients and saw what type of surgical intervention they were having to see what is their recurrence risk when it comes to bowel cancer, it turns out that if you were to do extended resection rather than just a small resection, your risk substantially reduces for all types of genes in which you find mutation. So clearly, something like this also helps you to identify what surgical intervention you need to do in the patients as well. Of course, this work came in Times of India earlier this year, and lastly, we collated all the information that we have so far across Lynch syndrome patients in India, and we ended up developing a website together with PLSD database, whereby if you come across a patient and you want to know what is their lifetime risk of developing cancer, you select which gene is affected, what is their age, whether they are male or female, and you get a beautiful profile. And based on that, you can start doing either screening or prevention work in these patients. So, to summarize, NGS Blitzkrieg, ultra-rapid, low-cost, high-throughput NGS assay, which can do MSI testing at scale. Missing link, we are only focusing on CRC. We are missing out 50% of the patients. We need to focus on the remaining 50% right now. MSI, it's the best biomarker you have to identify these patients. You get a substantial enrichment. Cancer prevention, it's not just aspirin. You can literally go bananas now. Which surgery? Well, you don't have to do small resection. You can now do extended resection, especially when you come to CRC, to reduce risk of metachronous colorectal cancer. And now we have the ability to do cancer risk prediction because we have India's data in it, along with global data. And not only that, finally we have founder mutations in India that can help you to do targeted diagnostics. With that, wonderful thanks to all the advisors, team members, collaborators, both scientific and research, and of course, the wonderful funders, GSBTM, who are sitting right here. Thank you.
Thank you. Oh, hello. Right. Thank you, Dr. Harsh Sheth, for that valuable address. And now I'll request Dr. Gunjan Kumar Mehta from GSBTM to present a memento to Dr. Sheth. Give a huge round of applause, ladies and gentlemen. Thank you. Thank you so much. Now may I request Dr. Ritesh Kumar, Assistant Professor, Animal Biotechnology, Gujarat Biotechnology University, to share their perspective on the topic, Innovation and Challenges in Immunotherapy. Dr. Ritesh Kumar is a molecular and cellular bioengineering researcher at Gujarat Biotechnology University, Gandhinagar, specializing in CRISPR-Cas9, genome editing, transgenic mouse models, and T-cell engineering. He completed his PhD at IIT Guwahati and postdoctoral research at Baylor Research Institute and UT Southwestern, and now focuses on innate immune responses in protozoan infections and genome-edited models for human and animal diseases. May I request Dr. Ritesh Kumar to take the stage, please.
Good afternoon, everyone. So today I'm going to talk about innovation and challenges in immunotherapy. Thanks to Dr. Mayank, he is somewhere. He has already explained very nicely about the challenges in immunotherapy. So I will talk more about innovations in immunotherapy. Before moving forward about my talk, I joined last year in Gujarat Biotechnology University after completing my postdoc from University of Texas Southwestern Medical Center and building my lab here in Gujarat Biotechnology University. The main focus of my lab has, if I can say, there are 3 pillars in my lab. One is the antigen discovery model, another would be the vaccine development, and third one will be the cellular engineering model. And for the cellular engineering model, we have an industry grant to develop a indigenous CAR T cell therapy development. And Dr. Mayank, I again cite him. He explained nicely about those tech. For antigen discovery, I am much more interested about immunopeptidomics and TCR engineering. As by training, I am a cellular engineer, and using those techniques, I am moving forward with the next generation vaccine development rather than the traditional or conventional vaccine development. I can say T-cell based or whole cell based vaccine development. And these vaccine development platform I am building on viral vector mediated or mRNA based delivery system for infectious diseases or for the cancer. Let's start with my presentation. I will put a word here, immunopeptidomics. I wrote it here as a game changer for cancer vaccine development. Why? Why this matters for vaccine and immunotherapy development? If you, based on your knowledge, if you think about the traditional vaccine development, there are two things. One is the known antigen, and another is predicted antigen. Based on that, all the vaccine development program will work. But immunopeptidomics, it provides lots of information, real information. If I talk about the real information, means the biologically relevant peptides which are presented on the MHC. We have to identify those peptides, and those peptides are really important. Why it is really important? Because those peptides, the MHC presented peptides, are biologically relevant and it elicits a strong T-cell immune response. I'm putting a word T-cell immune response. Why? Because when you talk about the therapeutic vaccine and when you talk about the immunotherapeutics, T-cell come in the picture rather than the B-cell or rather than the antibody. So another main focus is to identify that antigen. How? Let's talk about, this is the project thanks to GSBTM. When I joined GBU, I proposed this proposal to GSBTM, and thankfully they have sponsored this project without any data. They thought, okay, let them give some money and they will build those protocol here or project here in GBU. The committee realized, okay, this is out of the box. So this project I have proposed with Dr. Rakesh Rawal sir, he is somewhere here, and Dr. Kamal Mandal, one of the colleague at GBU. This is the immunopeptidomics guided HLA presented antigen discovery pipeline for immunotherapeutics development. So it's all about a technology. This technology can be used for infectious diseases in a pathogen model or in a cancer diseases or in a cancers where we can identify that HLA presented peptides that can be used for immunotherapeutics. Let's talk about the origin story of immunopeptides for better understanding. If you think, if you see the pictures, you can clearly see it's a model system, that's a HPV in a cervical cancer model system. When virus come inside, the virus antigens will be processed and will presented on the MHC, and that can be seen here, virus presented peptides, and that peptides can be used for vaccine development or for the immunotherapeutics development. In the similar scenario, you can look at the last corner of my slides. You can see there are a frame shift mutation that is very well known in the tumors, and we have seen lots of presentation since morning. People are talking about this frame shift mutation. From frame shift mutation, there are different kind of protein will form. That is, maybe if it is an insertion or deletion, that protein is not present in human system. But that proteins, once presented on their peptide fragments, once presented on MHC, that that we can say here as a tumor-specific antigen rather than the tumor-associated antigen. These tumor-specific antigens are not presented on any other cells, and that is the key of this technology. If we can tap that, we will be able to identify biologically relevant, naturally presented peptides on HLA molecule that are not present on any other cells, and that is the key to develop a therapy. How we can do this? Now, the it's a saying like immunopeptidomics is finding a needle from the haystack. But now, MS facility or mass spectrometry has been very well developed. We can identify those MHC presented peptides using this mass spectrometry methodology, where we use an antibody against MHC1 or against MHC2, pull out the complex, and then elute the peptide. When you pull out the complex, you pull out the complete MHC, means you are pulling out the MHC with the antigen, and now you put the acid, the antigen will be eluted. And these antigens pass through the cutoff filter, 10 kilodalton filter, MHC will be remain in the supernatant, and this will come down and run on the mass spectrometry, and then do the custom analysis pipeline, whatever you want to develop, you can identify the peptides. These peptides are naturally coming through if there is a frameshift mutation, these peptides does not match with any of the database, because the database is built based on the known proteome sequence of humans. Now, what you do a whole genome sequencing, or you do a RNA sequencing to identify the transcript where there is a mutation and translate those into a novel amino acid sequences, do a cleavage, make your own library that we can see here. We can say here is a custom MS-based database, and run the peptides that are not matching with anywhere, and that peptide will be the more specific, tumor-specific antigens, and that can be used as an immunotherapy. So what are the prerequisites for this experiment? We require an antibody. We cannot buy an antibody and do this experiment, because you require a milligram of antibody in one experiment. If I say milligram, let's say 8 milligram, 9 milligram, and if you buy one vial of antibody, it comes 1 mg. So you require own antibody purification platform in your laboratory, so that you have tons of antibody. I said tons of antibody means multiple vials of antibody in milligrams, so that you require multiple experiments, controls and tests and any experiments. So for this, we have established in our laboratory W632 hybridoma and purification platform, and we are able to purify sufficient amount of antibody. But for MHC2, we have an MTA with Perbrite. I got the sequence, put it in a clone and recombinant format. In a monoclonal antibody, we are purifying in a recombinant format. So now, these are the prerequisites to run this immunopeptidomics, and we have this capacity here in GBU, so we can purify lots of antibody. If you see, one of my PhD students recently joined 6 months back, because my lab is 1 year old, and she has established this protocol, and now we can see from 1 liter culture, we are able to purify 7 to 10 milligram of antibody per liter of culture for MHC1. And that we can see here, we validated this antibody. We can see heavy chain, light chain, and these antibody are properly binding on MHC1 of HeLa cells. So we are done with the prerequisite. And these are the complete workflow that I have shown here, how to identify the antigen. On the last corner, if you see, once the file has come from the MS, or output of the MS file, we have to run lots of in silico analysis like HLA binding prediction. Is it a real strong binder, weak binder, medium binder, from which HLA allele? Because if you think about the HLA presentation, let's say my HLA is HLA A and another one HLA is HLA B, the same epitope will not be presented on HLA A and HLA B. So we'll have to screen that in different HLA which epitope is presenting. Because we are running the sample with the one HLA type, but we want to screen with multiple HLA, that we do it in silico. Then we run with the MHC whiz pipe. MHC whiz pipe is a tool which predict where it is gonna, from which MHC binder it is, and is it toxic or not, then from which place it is coming, and there are lots of these analysis. So one of my a PhD student, what she said, he thought, okay, it's going to be very difficult. Once we have an MHC, once we have the file from MS and from the genome sequencing data, we make our own custom analysis pipeline. We find the peptides, but we don't know where is this peptide a truly MHC binder or not. We have to run lots of analysis. So it made an integrated pipeline in the lab. What she did, she connect all the available server at one place. So once you put the output file of MS and you choose you can see the options. You select and it will start giving the output file from that analysis, and that output file will be run on another analysis. So rather than doing this work for 1.5 months, you can do this all once MS is done. The prediction will be done in just less than 7 days or maybe less than 5 days. So this has been already developed, but we have not published it. That's why I put the mark unpublished. Now, if you want to see how the data looks like, this is Before moving towards the patient sample, I started optimizing this protocol or this technology on HeLa cells. For the, if you can see here the HeLa cells, you can see we are able to pull out 6,934 peptides from the HeLa cells. Here, for the mass spectrometry, we are using timsTOF 2 because other mass spectrometer does not have the capability to tap those peptides. We are in 200 nanogram of peptides. And 200 nanogram of peptides, if you run on other Orbitrap or any other system, will not get anything. So we need a timsTOF, and fortunately last year in India, in IIT Bombay, one timsTOF Pro 2 is installed. We don't have any other timsTOF anywhere in India. So thankfully, I don't have to send the sample to UK. Otherwise, I was planning to send the sample to UK. Now I'm sending the samples to IIT Bombay, and we are able to get a very good data. So this is one of the data from HLA. You can see here is 6,934 peptides, and most of the peptides, you see the length. Most of the peptides are around 9 to 15. That is the length of the MHC1 bound peptides. And from those peptides, once we checked how many of peptides are strong binders, we can find there are 232 peptides are strong binders. Then, this is the list of the peptides that we find with a very high score, and we checked from where these peptides are coming. For the vaccine candidate, it would be better if peptides are coming from the surface protein, so that their antibody will also be formed rather than the T cell. So that's why we check that. And then these peptides, we are going to test for antigenicity. Is it really antigenic or not? And then these antigenic peptides will further be tested in collaboration with NII and INSTEM in a patient-derived xenograft model. That's going to be, we are going to validate these peptides that can be that are truly immunogenic and has a huge potential to develop as a therapeutics vaccine. In another hour of my lab, we are trying to develop indigenous CAR T-cell therapy development, and I will not go in much more details because Dr. Mayank has already explained about its limitations. But based on these limitations, currently my lab is moving on this platform that we already have started on this domain, off-the-shelf CAR T-cell immunotherapy. If I say off-the-shelf, because in vivo engineering, what it does, it modifies. I think I'll just brush up this. I'll talk about the limitations of CAR T in just 3 words. Patient-specific complex manufacturing process. First, very high limitations. Second, it's a long production week, 2 weeks. Maybe after 2 weeks, patients are not ready to take the molecule. And the limited scalability. We cannot scale because it's a personalized. You have to take out the T cell from a patient. So one therapy will be useful for just only one patient. Each time you will have to build it. So then we thought, okay, why don't you move towards the off-the-shelf CAR T cell therapy where we can make a vial and that can directly administer it. If you see the prototype of this, we are making in a self-amplifying mRNA. Self-amplifying mRNA means we are using NSP, RNA-dependent RNA polymerase. When you give it the cells, it will proliferate and make multiple RNA copy. So our CAR construct, we are packing in a self-amplifying mRNA, and then these capsules are packing, we are packing in an LNP. And thanks to click chemistry, from the LNP, we are tagging the antibody, anti-CD3. So once we give this formulation to a patient that is directly, that is in the clinic, it will go inside, identify the T cell because this is a CD3 antibody and engineer it immediately, and that can be used as that is more scalable T-cell therapy. Half of this project was sponsored by Cell Immune and GBU collaboration, and this is our team. We are working with Kamal Mandal, he is assistant professor in GBU, and Dr. Abhishek Kahar, he is on the payroll of Cell Immune. He is a research scientist in my lab, and Arpita, she is working as on this project. So to summarize this, immunotherapy, what we talk about, this is a global race for the medicines of tomorrow, and everyone is running behind because we believe immunotherapy is the future. Now, immunopeptidomics, what I believe, and most of the immunology field, people believe that this is the key to develop next generation vaccine. If you have to identify the molecule, this is the key. And then CAR T-cell therapy, this is the revolutionary. We already have talked much more about details, and I would like to acknowledge my lab, my funding agency, GSBTM. This is the first grant we have received in the lab, and based on that, I built many things in 6 to 8 months of time. And DST, Cell Immune gives the grant for the T-cell engineering component, and GBU, these are my collaborators, Rakesh Rawal, Kamal Mandal, and this is my small lab group who is working on these projects. Thank you. Thank you.
Thank you, sir, for your valuable address. And now I'll request Dr. Nitish Dave to present a memento to Dr. Ritesh Kumar. A big round of applause, ladies and gentlemen. Thank you. And now may I request Dr. Gyanvel Mutharasu, Genix.ai to share their perspective on the topic AI-driven genomics in cancer, risk prediction to precision care. Dr. Gyanvel Mutharasu is the founder and CEO of Genix.ai, a technology-driven startup working at the interface of artificial intelligence and life sciences to build next-generation data and decision support solutions for genomics and healthcare. Please give a big hand for Dr. Gyanvel Mutharasu, ladies and gentlemen.
Thank you. Very good afternoon. I'm Gyanvel. I'm not a clinical doctor. I'll give a prelude. My background is in bioinformatics. I've done my bachelor's, master's, everything. Then I was working as a research fellow at Indian Institute of Science. Then moved to Finland. This is very relevant for the project. Bear with me for a minute. During my PhD, I had chance to work with many clinicians, diagnosticians, surgeons, especially in prostate cancer and glioblastoma, all around the world, Canada, US, UK. One day, we had an opportunity from Google and the NIH jointly working on integrating large volumes of genomic data into a Google data center, and we were to connect all the genomic data in US to bring it to Google Genomics infrastructure. There were 15 people around the world going to be part of that project. Luckily, I was one of them, analyzing thousands of sequencing data, building IT infrastructure that would stream it, also had an opportunity to analyze, extract patterns. That's story one. Second, it was late 2016. Our collaborator lab in Canada, there was a lady in her late 50s, walked into our partner lab clinic, and they've diagnosed stage 3, maybe stage 4. I'll tell what cancer it is. That's where the story is. They were not exactly sure about the origin of cancer. Then we asked them to send 5 ml of blood to our lab. We extracted the circulating tumor DNA in the blood, and we sent the report back to them saying, this is a prostate cancer. Were you following closely? It was a lady in her late 50s. No prostate, so no prostate cancer. The doctors were furious with us. And then we showed clearly this is a male DNA. We are not making mistake unless we have received wrong sample. We went back to the clinic, we went back and met the patient. We asked her to explain. Have we done any mistake? We verified everything. Then turned out she changed her gender surgically 20 years prior to the incident. And then there was a small, like a thin epithelial layer of cells left out, and that developed into cancer, started metastasizing into bone marrows and other parts of the body. Okay, that's only part of the story. You remember the Google story have told you that we were analyzing already thousands of genome sequencing data. From that, we have extracted many patterns. Now this castration-resistant metastatic prostate cancer is a separate group altogether. But when we started working, it was kind of new. We recommended there is no androgen in her body anymore, aggressive hormone replacement therapy, which means abiraterone is the typical treatment now. You don't have to do it. Enzalutamide, alternative, please go with that. That's our suggestion. That's the origin story. That's what actually we do in Genix. Basically, case by case, take the DNA, analyze it deeply. Let's say it's a typical story. That's how it looks like. And then we give that personalized treatment recommendation. Okay? And so let's say in a typical a rare disease diagnosis case, a pregnant couple visiting the clinic, the clinicians collect the sample. It comes to our lab. or our partner lab based on the geographical location where it is in the world. And then we extract DNA or RNA. We do usually whole exome sequencing is what we do, like 200x, 300x coverage. And then we analyze all the mutations in the sample. Until then, this is industry practice now in many parts of the world. Even in India, there are 50 companies, 54 to be exact, doing the sequencing and analysis, all that. It stops with that 4 steps what you see. Clinic visit, sample collection, DNA sequencing, variant calling, and some manual analysis done. A report goes back to the clinician. They do diagnosis or treatment planning. That is well established pretty much, though we are not doing as much as we could do. But our innovation at Genix AI is in the vertical column. We have a search engine that monitors not only PubChem and PubMed, but also 25,000 journal websites, archives, FDA, clinical trials, patents, every single website. Our search engine called Zealbots is monitoring. If there is a new research coming in, within an hour, that information is fed into our AI pipeline. And we have population-specific mutation database that is annotated on a daily basis, both through AI as well as using like human intelligence there to enrich that data. That's fed into 20 different pipelines we have. In this case, one of them is like a rare disease. That's where our company name Radiome Health Private Limited comes from, the rare disease and omics put together. And we make an gene investigation report, and based on the condition and status, we also have risk assessment AI models downstream. And a final report is composed and made available in the dashboard itself, because Genix is a SaaS platform. We give it to clinicians, hospitals, and other labs, and they could use that report to actually make an informed decision. We are a clinical investigation services company. That's what Genix AI is. Currently, our focus is helping with rare genetic disorders or cancer, of course, and also we are working with novel infections where like the lab test would take so much time. When I was working in Indian Institute of Science, my professor Srinivasan, actually he was hospitalized and then gone through pacemaker replacement surgery. Some infection went there, and he's no longer with us. It's like really unfortunate. I mean, that's like very strong incentive for us to use this technology and knowledge to give like timely detection of metagenomic sequencing. Right now, we are working mostly with hospitals, but we also work with pharma companies. We're not giving the data to them, rather they bring problems to us, and we analyze the data, what we see, those patterns we give as a feedback to them to conduct deep research or personalized combination or treatment strategies. It's what we do. Right now, we have hospitals using across the world, like Malaysia, Singapore, in the Middle East, and US also some of them. And in India, we've just started our operations recently. We are based out of Bangalore, and we are trying to establish a base here in Ahmedabad. Compared to those 54 companies in India, also there's 100 other companies around the world. The main difference is I have personally started working with genomic data from 2005 onwards. The past 20 years, whatever evolution of bioinformatics happened, we fed everything into our model. We fed everything into this mechanism, and we have, in our repository, we have 150 million research articles which are indexed, tokenized, reviewed, and fed into our system. That's like the biggest advantage. To train our model, we have used 1.5 petabytes of sequencing, annotation, and metadata. And it's not like fixed size. Every month, we are upgrading our models. We got access into India's fastest supercomputer to retrain the model recently in Pune. And I'm a bioinformatician. My team is a bioinformatics team, basically. That's where we bring value to all of you to use this information, this tool, to make an informed decision. There are many companies in the world. Some of them are working on the sequencing side, some of them are working in the analysis side, some of them are working on the annotation side, but we work on the investigation side of it, combining all of them together. For example, in our report, you might have seen report from many other companies, they give, BRCA2, BRCA1, PSA, PLA, where there is a mutation or not, that usually covers the coding part of the genome, exome, or maybe a gene panel. Specifically, you need to know, you have to give the theory to the bioinformatics company to give you expected result. What if it is not covered? If you take, a good gene panel, it shouldn't cover only like one gene like BRCA2. Usually on the treatment, there's going to be other genes involved in the treatment outcome that we are completely missing out. If you take a comprehensive coverage, we should cover the coding region, non-coding region, at least the promoters. We should see at each gene what is going to be the contribution, which article that evidence is taken from should be there. And right now, the way bioinformatics analysis is done, it depends on the expertise of that one particular student, maybe intern in some company. It's not like systematically documented. There is no way for us to go back and then see what threshold has been used on a particular tool or algorithm. That's why we automated most of this process in an auditable manner. It's HIPAA compliant, GDPR compliant, and we are also going for the clinical decision-making system approvals, like 513 de novo in FDA and CDSCO, all that. Just to prove that we have a system rather than a software, rather than a tool, rather than a data or a manual analysis. That's where we come from. Yes, and we also provide this kind of recommendation with evidences, with suggestions. There is this article, it suggests there is this FDA approved or clinical trial currently going on. So maybe you can consider. Of course, all the doctors in India are like overworked. So much work already. How much time you can spend on like reading hundreds of articles every single day? At least if the system gives, okay, doctor, there's like a 10 article I found it recently, seems relevant to you. Please take a look at this. That's what our system does, saving time, okay? So that's where we are, actually on time. If there are any questions, please reach out to me. Thank you so much.
Thank you so much, sir. I'll request Ms. Pankti Mistry from GSBTM to present a memento to Dr. Yanavel. A huge round of applause, ladies and gentlemen, for him. Thank you so much. May I now request Dr. Siddharth Shah, Head and Neck Cancer Surgeon, Zydus Hospital, to share their perspective on the topic, Catch Them Early, Treat Them Early. Dr. Siddharth Shah is the lead head and neck cancer surgeon at Zydus Hospital, Ahmedabad, a pioneer of scarless transorals in India. He specializes in complex and robotic cancer surgeries with an emphasis on preserving patient quality of life. Yes, a big round of applause for Dr. Siddharth Shah, ladies and gentlemen.
Thank you. My condition right now is like Amir Khan in movie PK when he enters the Earth globe. So I am like that in this forum. Okay, so I am like a kid who always cribs about his problems and doesn't want any solution. So I'm going to here discuss my problems and maybe seeking solutions from you. So that's why I chose a topic like from anatomy to biology, seeking answers in proteomics. Next slide, please. Okay. So I have a distinguished senior professor with me, Dr. Somesh sir. Sir, please guide me if I am anywhere missing out any topics. So most importantly, I'm a head neck cancer surgeon. So my basic talk will be more into head neck and especially in oral cancer. So what we want is ultimately in cancer treatment is outcome. We want patient to do good, do best in their outcome irrespective of the stages. We want to improve their survival. Over last decades, probably we have improved on technology. We have improved on diagnostics. We are diagnosing very nicely early, picking up cancers with technology. But probably the treatment outcome or the response is still not that marginally improved, or is marginally improved, not drastically improved. So somewhere we are still not able to understand the biology behind this tumor, and this is where you probably help us define it, probably help us in treatment and guide us in how we should progress further. So how this behavioral pattern of cancer understanding will help us is kind of tailoring a personalized care for each patient rather than one size fits all, and also to predict their tumor response, recurrence pattern, as well as the survival prediction. So probably somewhere what we want is under this category or idea. What are our present limitations? Our TNM guideline is more of an anatomical model where it just tells me that tumor is reaching here, there, so it's probably this stage. But it doesn't truly predict the behavior pattern of the tumor. Similar histology, similar staging, but there are different outcomes. And so we do need some biology-driven product models where we can personalize the treatment. In a way, we want to predict the future like this, man. So in head neck, when we say we have local control, regional control, and distant metastasis or distant spread. So this is the area where we kind of play around. And what is important is like the local control. In head neck, there are various subsets in the body. So head neck probably has the highest failure, is more of a local failure, followed by regional failure, and followed by distant failure. So what we want is, how can we improve our outcomes by giving best control on the local area or the primary site area. And that probably, because almost 20 to 30% patients will have some recurrence in the local area, same area. So what we have and what we probably are moving towards in the present era, we have only clinical and radiological staging to guide us for treatment. We have cross histopathology to tell us whether our margins are clean, clear or not. And we ink the margins and they tell us the morphology of the margin. That's it. So what probably might help us future is the molecular margin, genetic risk stratification, proteomics, and the personalized therapy surveillance. Now my first problem is like this, my first cry. I am not able to predict my local failure. I am not able to prevent my local failure. Why? Because I am not sure about my molecular margins. Whatever we study is the morphological margin, anatomical margins. So there can be genetically altered cells at the margin of my resection which may behave bad. There is a phenomenon called field cancerization. That's all you might be aware about because of the tobacco addiction and all. And that's why probably even if I'm boast about my margins, like I've done my live biopsy, that's what people say, and they are 1 kilometer away, but still patient fails in 20% cases. So how can I do or predict my molecular margins? And how can I do that? So at GSBTM, I had initiated a collaborative project of assessing a molecular margin. We have got some breakthrough, but we are still hindered at some place. Prankti might throw light on that. And whether this molecular margin evaluation has any role to predict or plan for adjuvant therapy. So we have this information with us about the mutations of the margins, the markers, all the receptors, which helps to we know that they are responsible for margin recurrence and survival impact, but right now we don't have anything to assess them. And that's what we wanted to do as a study, and we are somewhere midway stuck, assessing the molecular margin on table, on surgical table, that we can find out if we need to clear it further or not. So if somebody that's what I was told, that it's a forum where probably I can throw my problems, and somebody might be here to give me a solution. So we don't have any standardized molecular panel. So next problem is my regional or distant metastasis. And these are certain markers which are actually already identified, but there is no uniformity and there is variability into this. Next is tumor behavior. I don't know how my tumor is going to behave. A simple example, when there is an advanced tumor, we do give them chemotherapy to downstage this tumor, so that that patient becomes more comfortable for surgery. But the same tumor behaves in different patients differently. Some might have 10% response, some might have 20% response, some might have 70% response. We don't know which tumor is going to respond equally. I mean, how the tumors will go with this medication or not. So there is variability of this tumor behavior. And so there is no tailoring that we can do. We cannot plan, okay, exactly we should go with this SOP. Primary versus nodal versus recurrent tumor profiles also differ. We don't know whether we should study a single biopsy or an entire whole biopsy specimen, because there will be a gross heterogeneity in tumor biology in the entire tumor. Also, we need to understand which tumors may be more radiosensitive or not, and tumor aggressiveness. So which molecular profile should guide us treatment to understand this tumor behavior? How can we understand this tumor more better? So we have this tumor biology markers like proteomic markers, gene expression signatures. By and large, we know that they will help us to understand. But in clinic, how do we incorporate them in our day-to-day activities or into day-to-day planning and treatment care? This is where probably we need some light or hope from you. There's a lot of genetic variability. We know that P53 mutation is responsible for poor survival. The Notch1, PIK3CA, they are all markers which have been identified in head and neck squamous cell carcinoma. But the same mutation has a differential survival outcome. There is a tumor heterogeneity effect, because the same product, probably the cisplatin or maybe anything, will not give 100% response. There will be some tumor which may respond to tumor, and the part of the tumor may not be at all touched upon. There will be resistance, tumor resistance, medicine resistance. So how these tumors develop this resistance to medicines? So this is certain things which are probably we need to understand. How can we predict early failures? Patient has been treated completely, surgery done, radiation done, chemotherapy done, everything. Now we keep patients on follow-up. We just can see the patient clinically. We can do at the most evaluation like imaging, which is more of an anatomical and functional imaging, which is a recent. But molecular imaging is not possible. So how can we predict those early recurrences? How can we pick up those recurrences so that probably we can treat them earlier? How can we predict an individual patient will behave that way or maybe behave worse in future right from the beginning of my start of the treatment? So how can I understand the biology of this aggressive tumor? And so we do understand there is a segment of therapy coming in future is gene therapy. So probably one way the answer could be future is a gene therapy to prevent all these problems. But what can be done interimly as of now? So again, coming from anatomy to biology, now I'm seeking is biology to anatomy. And my previous speaker very well alluded about the AI, role of AI coming up into the oncology, into the omics. So probably somewhere it will help us to understand the tumor treatment, the tumor resistance pattern, the recurrence pattern, and predict this recurrence and improve the survival. So what I envision is omics-driven cancer care, where surgery will be guided by molecular margins, adjuvant therapy by the tumor biology, surveillance by liquid biopsy, and outcome by integrated omics. So probably we need to have a biological TNM model for the patient care. Thank you.
Thank you. Thank you so much, sir. I'll request Mr. Harmadan Gadvi from GSBTM to present a memento to Dr. Siddharth Shah. A big hand, ladies and gentlemen. Thank you. Thank you so much, sir. All right, ladies and gentlemen, after a knowledge-enriching session, we are quickly moving towards the next session, the technologies from the state. In this session, we have briefly tried to cover the technologies or processes developed with state funding and the commercialization efforts that have gone behind these technologies. To begin the session, may I invite upon the podium Dr. Amit Shad, Assistant Professor, NIPER Ahmedabad, to address the audience. Dr. Amit Shad is an assistant professor in medicinal chemistry at NIPER Ahmedabad. His research focuses on the rational design and synthesis of novel small molecules, such as anti-leukemic agents, contributing to the institute's pipeline for new drug discovery. Can we have Dr. Amit Shad on stage, please?
Good afternoon, everyone. This is Amit. I'm from NIPER Ahmedabad. I work in anti-cancer drug discovery. And the target that we have chosen to work upon is pyruvate kinase M2. And the target that we have chosen, the reason being that it is one of the most important target in the glycolysis. And after its mutation, the cancerous the pyruvate kinase M2 migrates to the nucleus, and the cancerous cells turn extremely malignant, and the proliferation takes place. How to move this slide, please? Next. Pointer is there? Yeah. So this is a brief background about the lung cancer. So what we, in this presentation, I'm going to present upon is that we have developed a series of the sulfonamides, derived pyruvate kinase M2 activators, which act as potent anti-lung cancer agents. So this is a brief background of the lung cancer, that it is second most frequent type of the cancers, with 18.4% of all cancer-related fatalities, and 5-year survival rate in case of lung cancer is only 28%. In India, it corresponds to almost 5.5% of all cases and 8.1% of cancer-related deaths. The annual treatment is phenomenally very high and unaffordable for the common man. So small cell lung carcinoma and non-small cell lung carcinoma are the most common types. So these are the different stages, which I'm going to skip. And several celebrities who get infected with it, they might get cured, but we must also be thinking about the common man who gets cancer, infected with it, but is not having the opportunity to get it treated, to get the cancer eliminated. We need affordable health care also. And the existing treatment line that is existing may be chemotherapy, targeted therapy, immunotherapy, or anti-angiogenic therapies. But all of them have, although they have added certain months to the life of the patient, but complete cure is required, new agents are being required, and each of the treatment line is having its own side effects. And the most common one is the chemoresistance. So of course the chemotherapy is the main line of the treatment in case of the cancer. So why do we need, although we do have a number of drugs that are being prescribed in case of the lung cancer, maybe paclitaxel, docetaxel, cisplatins, and several others, but these are maligned by the severe chemoresistance and side effects, which even make the life of the patient extremely complicated. So this necessitates the development of new chemotherapeutics. According to the nature genetics, it is very much required that sometimes the molecule that we are trying to bring to the fore might not act as a drug, but its co-administration with the existing line of the chemotherapy might help in improving the patient outcomes, and we may be able to add years to the life of the patient rather than months. So this is the global lung cancer market size, and it is being believed that its market size is expected to increase from $29.5 billion US to $86.5 billion US, with a compound annual growth rate of almost 11.5%. So using our computational tools and after going through several molecules which are being pursued in the clinic against lung cancer, we had almost a database of 11 to 1300 molecules. We used some computational methods to figure out that which type of moiety is presently being focused upon for the treatment of the lung cancer. We found that sulfonamide moiety is coming time and again to us, and it was found to have a reasonable interaction with the pyruvate kinase M2. And when we looked into the literature, we found that there are several drugs which are having sulfonamide moiety, and some of them are enlisted over there, and they have a huge market size. So taking inspiration from that and certain molecules in preclinical trials, etc., we ventured upon to develop certain molecules which were based upon sulfonamides. And sulfonamide is the moiety having SO2 and NH moiety inside the molecule. And considering these type of molecules, we try to develop a library and screen them against the pyruvate kinase M2, which is found to be having high concentration in the malignant lung cancer cells. So as of now, when we look into the literature, we found that till date there is only one molecule, TP1454, which is there in phase 1 clinical trial, and it is being given with docetaxel against lung cancer. And it is found that this molecule locks pyruvate kinase M2 in the tetrameric state. Once if it is in the tetrameric state, it cannot migrate to the nucleus and the cancerous cells cannot divide and multiply. So focused upon the prior literature and the molecule that we had sought inspiration from, we tried to develop a library of the sulfonamides. Since our lab works on pyruvate kinase M2 modulators, we had number of publications on pyruvate kinase M2 in versatile journals. So in this work particularly, which was taken up in our lab, I have skipped several slides in between. We designed, synthesized, by means of computation, we designed close to 100 plus molecules. We had sought inspiration from 1100 molecules that were doing very good in vivo, as well as some of them were in the preclinical trials. We found that yields were also quite reasonable through a multi-step synthesis, about 52 to 70% yields. The molecules were causing G2 phase arrest in A549 cells. They were causing early apoptosis in the cells, and AC50, that means that the activation concentration was found to be almost 30 nanomolar. Through a series of bioactivity assays and surface plasmon resonance studies, etc., we could find that molecule is having reasonable interaction with the pyruvate kinase M2. And we also did preliminary pharmacokinetic studies for the molecule that was being developed in our lab. And we found that the log P was that of the means around 2. It was orally bioavailable, and it had good plasma protein binding, plasma stability, and 3 metabolites were found, and they are probably acting as an antineoplastic agent inside. And Cmax, Tmax, T-half, etc. were all found to be comparable to the standard, and we think that this molecule has potential or legs to move down the pathway of the drug discovery. But as of now, we are yet to validate this in animal model, but the preliminary DMPK studies are done, and molecule is found to be orally bioavailable. We are at present TRL level 5, and we are planning for the mouse xenograft model studies, and we are also developing in the similar way. We patented this molecule and this class altogether, and the results were later on published in RSC Medicinal Chemistry. The follow-up paper on the drug metabolism and pharmacokinetic was also published subsequently. So we are also having certain other molecules in the pipeline that work against pyruvate kinase M2. And this is the general trend that we follow. And we do have molecules that work well and have been validated in animal models, nude mice models, and we closely work or integrate with the Department of Pharmacology, Department of Biotechnology, and faculties from the Department of Biotechnology like Dr. Amit Pandey, who is there with me, and So these type of studies that we also have the similar type of pyruvate kinase M2 inhibitors developed very lately, and the molecule was found to be very potent, and very lately we published those results in Journal of Medicinal Chemistry, ACS. One surprising fact I would like to bring to the kind attention of the August gathering is this, that the molecule that we very lately published as PKM2 inhibitor against pyruvate kinase M2 in JMedChem, it was not only found to reduce the size of the tumors in azoxymethane-induced mouse model, but it was also found to restore the microbiota of the gut after treatment. So Journal of Medicinal Chemistry and its editors, they applauded our work. They said that this might be laying down an extra care from the side of the medicinal chemists, that we should not only be developing the molecules which are having therapeutic potential, but the gut flora, which usually gets disturbed because of the heavy chemotherapy sessions and very low immunity that the patient goes through down the line, might be a stepping stone for future type of therapies. So these type of molecules we are also pursuing currently, and this is kind acknowledgement to the all funders, including GSPTM, ICMR, DBT, Department of Pharmaceuticals, and last but not the least, NIPER Ahmedabad. I thank everyone for the kind attention, and if you have any queries, I am available and you can write to me out. Thank you so much.
Thank you. Thank you so much, sir. I'll request Dr. Gunjan Kumar Mehta to present a memento to sir. A big round of applause, ladies and gentlemen. Thank you. Thank you so much. May I now have the privilege to invite Dr. Frankie Shah, GCRI Ahmedabad, for the next address. Dr. Frankie Shah is a senior scientific officer leading the Cancer Biology Department at the Gujarat Cancer and Research Institute, GCRI. With a PhD in life sciences, she oversees research focused on cancer biomarkers and molecular diagnostics to support the Institute's clinical research goals. A big hand for Dr. Frankie Shah, ladies and gentlemen.
So, good evening, respected dignitaries, colleagues and friends. Today I am going to present my work on HBOC and how integrated genomic technologies uncover the biology behind the disease. So let's have a brief introduction. Hereditary breast ovarian cancer, or HBOC, is a genetic condition that makes someone more likely to get breast, ovarian, prostate, pancreas and other cancers. And the major gene involved associated with HBOC is BRCA1 and BRCA2. The presence of BRCA1 and BRCA2 predispose an individual to develop disease if BRCA1 or BRCA2 present in the genome. So there are presence of BRCA1 and 2 mutation is quite less rather than patient without BRCA1 and BRCA2 mutation. And this led us to know, to have a question that there are certain hidden germline genomic vulnerabilities beyond BRCA1-2 mutation which contribute to HBOC risk. So here we are trying to identify the underlying molecular mechanism in HBOC with the technologies microarray and whole exome sequencing. So here we have used copy number CN-LOH and copy number variation using RSEGH plus SNP kit of Agilent. So in this study, we have included 44 HBOC patients, and This platform actually analyzes copy number alteration as well as copy neutral loss of heterozygosity involving 1,70,000 distinct biological probes affording a resolution of 5 to 10 MB for CNLOH. So after QC correction, we found that there are various CNVs present in these patients. After removing the general CNVs, there are rare CNVs we found, that is 2.86% from this patient. The most prevalent chromosome region exhibited copy number gains were identified in 6q and 22q, 9p, 13q and 21q. The most frequent affected regions with copy number loss were observed in 9p and 5q. And chromosome 6 and 22 exhibited the highest incidence of structural CNVs. There are certain recurrent CNVs in certain patients also. So these CNVs comprised approximately 290 genes, which are mainly from coding region, microRNA, LNC RNA, SNO RNA, and pseudogenes. So when we analyze these CNVs, we found that there are certain genes which are associated with cancer, and they are tumor suppressors, oncogenes, genes involved in genomic instability, and metabolic and oxidative stress related genes. So the identified alterations affect tumor suppressive pathways, oncogenic signaling, and chromosomal stability. So now coming to the loss of heterozygosity, this is the distribution and frequency of LOH. So chromosome number 6 shows maximum LOH, followed by chromosome 1, 20, 2, 8, 9, and X. More than 50% of the patient showed the CNLOH. So this CNLOH showed the regions which are associated with DNA repair and maintenance, cell cycle control, and chromosomal segregation, apoptosis and cell survival, and transcriptional regulation in cellular metabolism. So the presence of CNLOH without changes in copy number suggests allelic imbalance. And there are certain key cancer genes were found in this LOH region, like Notch, MRE11, PALB2, ERCC4, and TP53. So this microarray will give you the gain, loss, and so they will give copy number variation, but they do not tell you the changes in sequence. That's why we have analyzed the samples using whole exome sequencing. This is the workflow of whole exome sequencing. We use Ion Gene Studio S5 sequencer, and variants were analyzed and classified according to ACMG guideline. So our whole exome sequencing result showed that there are 23 unique genes and 40 total mutations. From these genes, there are 15 pathogenic mutation, 7 likely pathogenic mutation, 12 benign or pathogenic, and 6 were risk factors. And there are certain genes which show loss of function, and they are frame shift deletion, typically lead to premature protein truncation. The affected genes are CRBBP, NBAS, GLY1, OCA2, DNAH9, NSD2, and SLCO2A1. So missense variants were predominant in our cohort, and they contribute to 70% of identified variants. This table shows the patient number, identification number, along with their genes affected. And these genes include tumor suppressor CREBP, genes with oncogenic and driver evidence that are GLY1, NSD2, LGR4, and genes where germline variant are known to modify risk, that is NQO1. There are many genes have important role in tumor biology, microenvironment, thrombosis, immune response to DNA repair signaling, but are not established classical germline cancer predisposition gene. So this sequencing result tell us that the genetic under the HBOC is very diverse, and NQ1 is the most frequently mutated gene, suggesting population-specific hotspot. And 15 confirmed pathogenic variants identified, and they required immediate clinical action. So we have integrated this microarray and whole exome sequencing analysis data to enable. So this combination of these two technology will provide the complete germline profile of patient, enabling detection of pathogenic sequence variants as well as copy number changes and copy neutral LOH, and reveals mechanism of gene inactivation relevant to hereditary cancer risk. So this is the real patient data. I will share 2-3 patient data we have analyzed using these two technologies. So this is patient number 1, was diagnosed with breast cancer. The molecular subtype was luminal B. The patient was presented with advanced stage, and the current status of patient is alive. So this is the data of this patient, where we found that there is gain in 3 chromosomal region, that is 8q 24.3, 19q 13.43, and 21q 22.3. So this gain results into DNA repair stress response, epigenetic regulation, DDR pathway associated genes, and RNA metabolism and cellular stress. So germline copy number gain suggests a background genomic instability and epigenetic dysregulation predisposing to cancer development rather than acting as a direct driver. Our whole exome sequencing results show that there is presence of variant NQ01. So its functional effect is the enzyme loss of enzyme activity, and its cancer relevance is it is associated with oxidative DNA damage and breast cancer risks. So whole exome sequencing reveal modifier variant affecting redox homeostasis and DNA damage tolerance. So here in this patient, we can see that there is CNV contribution and SNV contribution. So, and these are the pathways, DNA damage and repair pathway, where RECQL4 gain was there and increased oxidative DNA damage. Stress response shows that HSF1 gain and redox imbalance. Epigenetic control pathway includes stream28 gain and indirect transcriptional deregulation. And cancer susceptibility shows structural variation and functional polymorphism. Should I? So give me 5 minutes more. So the combined presence of germline copy number gains affecting DNA repair and epigenetic regulation together with functional CNVs, SNVs, impairing oxidative stress defense supports a polygenic modifier-driven model of hereditary breast cancer. This is a TNBC patient, and the patient was came with early stage, but survived for only 1 month. So here the chromosomal variations in this. Here you can see that there is gain in 5p 15.33 and 8q 24.3. So these two regions are associated with cancer, and they associated with particularly 8q 24.3 associated with cancer hotspot, and it has an oncogenic potential and likely most significant finding. Its whole exome sequencing result showed that there is a presence of a frame shift deletion in KLH-L41 gene. So this frame shift deletion will causes premature stop codon, which is associated with congenital myopathies, and this is very rare in population, and it shows a carrier status. So combining all these data, the primary germline finding of cancer relevance is 8q24.3 gain, and whole exome sequencing variants may indicate rare genetic predisposition. So I'll skip the data, but in general, this HBOC is a very heterogeneous disease. So it arises from multiple genetic, epigenetic, and other molecular mechanism. So what are the clinical and translation implications? It explains hereditary risk beyond BRCA1-2 and suggests BRCA negative HBOC is not genomically silent and supports expanded germline testing. Further, this result uncovers germline 2-hit events, inherited variants combined with CNLOH contribute to hereditary cancer susceptibility, and integrated blood-based genomic analysis improves risk interpretation. Apart from that, we have a lot of work done in HBOC, and these are the publication. And I'm very much thankful to the funders, and this is my team. And I also would like to thank the patients and their family also. Thank you very much.
Thank you so much. I'll request Dr. Nitish Dave to come upon stage to present a memento to Dr. Frankie Shah. Give a big hand, ladies and gentlemen. Thank you. May I now have an honor to invite Dr. Vivek Tanavade, Ahmedabad University, for sharing his knowledge with us. Dr. Vivek is an associate professor and associate dean at Ahmedabad University, focusing on cancer biomarkers, drawing on stem cell biology training at Johns Hopkins University and prior PI experience at A-Star Singapore. His research centers on mesenchymal stem cells and non-invasive salivary exosome-based diagnostics for oral cancer. Can we have Dr. Tanavade on stage, please?
Yeah. Thank you. I'll try to keep to time, considering that we're already running late. So, a disclaimer, I no longer work with mesenchymal stem cells or stromal cells. That's my past. I now focus exclusively on head and neck cancer ever since I've come to Ahmedabad about 9 years ago. The reason is this is a local problem. Right? So much of this work was done by Kanisha, who is an ICMR woman scientist in my group, who is wearing a red top, and Shreya with a yellow top, who is currently a PhD student. So just to introduce, not that this audience needs introduction, but this is a problem for Gujarat. Dr. Siddharth Shah explained this extremely well in terms of the challenges that are faced by clinicians. We've been focusing on dealing with chemoresistance and looking at salivary biomarkers that can predict chemoresistance, but I'm not going to talk about that today. Today I'm going to talk about the project that was funded by GSBTM, and one of the ways to really reduce chemoresistance is to reduce the dosage of the drug itself. If we can achieve that, then the resistance would also go down. So in collaboration with Dheeraj Bhatia's lab at IIT Gandhinagar, we designed DNA tetrahedrons which can be encapsulated with different drugs. You can also functionalize them with different molecules. Now, why DNA tetrahedrons? So the original project that we wrote for GSBTM actually had nanoliposomes. That's what we had, and that got funded. But then when Dheeraj and me started talking, we realized that this is a much better way to encapsulate drugs compared to nanoliposomes. The reasons are that DNA, being a natural material, easily taken up by the cells. We're all familiar with transformation. It is relatively easy to have, especially drugs like cisplatin, which are smaller molecules, which can be encapsulated into these tetrahedrons. And it is also very easy to functionalize. If you want to add linker spacers, you have the sequence is known. So with all these advantages, we went back to GSB team and said, can we switch this to DNA tetrahedrons? This shows you the characterization of the TDs with cisplatin. We used a number of different methods, AFM, FTIR. The size was around 18 to 20 nanometers. And what I really want to show here is the release. So the last line plot shows you that especially if you look at the red line, you'll see that at pH 5, the TDs plus cisplatin, and including at neutral pH, there's a very nice sustained release of the drug from the TDs. When we looked at the functional status of these cells, so we did all this work using a human cell line called as OECM1. So this is a human oral squamous cell carcinoma cell line, commercially available, derived from a patient, and very much the tumor that we see here, which is in the buccal cavity. Encapsulating the TDs with cisplatin reduced the concentration. The IC50 came down from somewhere around 500-600 to about 200. It also induced apoptosis. This is an XN PI assay. As you can see, the TDs plus cisplatin gives you a much higher number of annexin positive PI negative cells. We try to look at what is the mechanism of this cell kill with apoptosis, and what we observed was that there is a very high amount of ROS, or reactive oxygen species, that are being generated. What you can see here is that especially in the TDs plus cisplatin, so this doesn't happen so much our comparisons were always with TDs alone and cisplatin alone. So it's not only bringing down the concentration of the drug, it is also achieving an effect, a biological effect, that a drug alone can't achieve. So when we encapsulate cisplatin in TDs, we're getting a lot more ROS, as we saw by DCFDA staining. We also looked at the migration and invasion of cells. What you see here is a classical scratch assay or a wound healing assay. And after 24 hours, you can see that in the control, you have a lot of wound healing happening. The cisplatin and the TDs plus cisplatin reduce this migration. On the right side, what you see is a phalloidin assay. So this is a spheroid assay where you look at the invasion of these cells. In the control OECM1 cells, you see that there is a tight nucleus and a lot of spread around. Especially when you look at the merged, you can see that very well. It is still there in the TDs, and also to a certain extent in this cisplatin alone. The spread is much lower when you encapsulate this cisplatin. Now remember, this is happening at less than half the concentration of the cisplatin alone. Right? And so when we did all of this, we also wanted to check what happens. All of this is fine. This is in vitro. What really happens in vivo? And so in my lab, we had developed a human oral cancer mouse model. This is a xenograft NOTS-KID mouse model that we work with ICER Pune. And by about 26 to 28 days, we can get very nice tumors. So the way we do this is we inject these OECM1 cells into the roof palate of this NOTS-KID mouse, and it is a human tumor, and histopathologically very, very similar to what we see in the patients. So it's a good model system to then try and test out our drugs. And what we can see here is that in the control, you get a very nice tumor. Cisplatin does cause a reduction in the tumor, and TDs plus cisplatin also causes a reduction in the tumor. But what is more interesting to me is that if you look at the histopathology, you will see that in the TDs plus cisplatin, it looks a lot more normal. It's less nucleated, better organized, compared to even the cisplatin. And there was no toxicity that was achieved or observed in the liver, lungs, kidney, brain, spleen, heart, etc. So to sum this up, encapsulating a well-known cancer drug like cisplatin into a novel scaffold like DNA tetrahedron can not only bring down the effective dosage, but it can also result in biological phenomena that are not readily observed with cisplatin, which give you a better kill of the tumor. So I'd like to thank Dheeraj, who was a co-investigator on this project at IIT Gandhinagar, and we worked with his lab. He has the expertise for the TDs. Dr. Suraj Ingle at IISER Pune, who helped us with our xenograft model, and of course GSBTM for funding this. Yeah. Thank you.
Thank you. Thank you so much, sir, for your valuable address. I'll now request Ms. Pankti Mistry from GSBTM to come up on stage to present a memento to sir. A huge round of applause, ladies and gentlemen. Thank you. Thank you so much. Now may I invite upon the podium Dr. Reena Rajput, Professor, Department of Biotechnology, Naipur, Ahmedabad, to address the audience. Dr. Reena Rajput is a professor in the Department of Biotechnology at Naipur, Ahmedabad, working in immunology and cancer biology with a PhD from NCCS Pune University and postdoctoral experience at B.V. Patel Bird Center and Naipur, Ahmedabad. Previously, she served at the Institute of Advanced Research, Gandhinagar, where she held key academic and research leadership roles in biotechnology and bioengineering. A big round of applause for Dr. Reena Rajput, ladies and gentlemen.
Thank you so much, GSBTM, Dr. Anusuya, Pankti, for giving me an opportunity. So I would be discussing a brief on a GSBTM funded project, which my student just concluded. And in this project, what we have tried to do is we have tried to reposition a very, very known antibiotic, which is doxycycline, and we have tried to establish it as an immune checkpoint inhibitor. So when we talk about cancer, TNBC, that is the triple negative breast cancer, are difficult to treat. There is a lot of heterogeneity in the triple negative, ERPR positive, HER2 positive. But then when we talk about TNBC, it poses challenge, particularly when we talk about developing the immune checkpoint inhibitors. So we took this problem of TNBC and then we tried to see what are the challenges posed by TNBCs. So when we talk about any breast cancer, it is a heterogeneous mass wherein a lot of immune cells infiltrate. And the fate of the tumor would be decided not only by the growing and progressing tumor, but also by the immune cells that have infiltrated. And one of the very key and pertinent cells that infiltrate the tumor microenvironments that basically orchestrate the entire network of tumor outcome are the macrophages. These macrophages can generally exist as two forms. One is the M1 macrophages, which is the anti-tumor macrophages. So when the macrophages are infiltrated at the tumor site, they generally would differentiate into an anti-tumor M1 macrophages, which would then prime the T cell, the T helper 1 cells and the CD8 T cells to cause tumor regression. However, tumor secretome primes majority of these infiltrated macrophages into the M2 macrophages, which in the tumor microenvironment we generally term as the tumor-associated macrophages. And these tumor-associated macrophages are responsible for all of the events that follows in cancer metastasis, neovascularization and progression to the secondary sites, which remain one of the major causes of mortality. So this project centers around utilizing the potential and reprogramming of macrophages and to develop a very novel and small immune checkpoint inhibitor. So moving further, when any of the immune cells at the tumor microenvironment, if they are at all able to kill the tumor cells, naturally the dead and the dying cells would release a lot of sequestered metabolites. And one of the very, very important molecule which is secreted by the dead and the dying cells are the extracellular ATP or the ATP that accumulates in the extracellular environment. Now in the tumor microenvironment, particularly in the advanced stages of cancer, The immune cells as well as the cancer cells express the family of enzyme receptors which are termed as actonucleotidase. These actonucleotidase are CD73 and CD39. And these actonucleotides, they basically help in converting ATP into ADP and subsequently into AMP and adenosine. What is the problem with this adenosine accumulation and ATP metabolism is that it renders immunosuppression. So we took this as a major problem and as a major driver for our work. And then we thought to repurpose some of these small molecules as a novel immune checkpoint inhibitors. And one of the major inspiration came from this paper by Lisanti, where the daughter of this person, Lisanti, advised why not give antibiotic to the cancer patient. And with this idea, they tried antibiotics and they showed because these antibiotics are able to target the mitochondria, we can use them as the anti-cancer therapy. And then of many antibiotics, we did a lot of literature survey, and then we thought doxycycline could possibly be a very good target, which can be repurposed as an anti-cancer immunotherapeutic molecules. So we moved on. We screened a lot of antibiotics, and then generally, as expected from the literature survey, doxycycline was able to kill the tumor cells very well at an IC50 value of around 24. So we did an initial screening just to see the anti-cancer potential, and the doxycycline was able to cause the tumor killing, induce apoptosis, cell cycle manipulation and others. But as I said, at the immunology lab, my focus is mainly on this important cell that are the macrophages. So we wanted to see what is the impact of this doxycycline on the macrophages. So as I said, M1 macrophages are the macrophages that have anti-tumor activity, while in the tumor microenvironment, as we can see in the panel marked in red, we have an increased abundance of the M2, which are the tumor-associated macrophages. And when we treat these macrophages in the tumor-mimicking condition with doxycycline, we are able to see that they revert back into an activated macrophages. And so we wanted to validate it in the preclinical model. So we treated the 41 breast cancer bearing animals with doxycycline. And though we see a very good improvement in the immunophenotype, that is the macrophage M2 were decreased very significantly, even the T cells were getting activated, but it was not translating into a tumor regression. So then we brainstormed what could be the possible reason that there are a lot of immune activation, but still we are not able to get the tumor regression as expected. So then we screened the secretome of the tumor microenvironment for almost many of the metabolite. And to our surprise, the tumor secretome was secreting a lot of ATP. And then as the tumor was progressing, we observed that this ATP was metabolized into an immunosuppressive adenosine. So then we thought, can we use the antagonist of these ectoenzymes, that is CD73 and 39, along with doxycycline, so that we can have a better repurposing of doxycycline as an anti-cancer and as an immune checkpoint for blocking or inhibiting CD39 and 73. And as we can see here, when we combine doxycycline with these antagonists, we see the positivity of these ectoenzymes decreasing very, very significantly and synergizing very well with doxycycline. Then we moved further, and then we did the immunophenotyping for the M1, M2 frequency, and as expected, this strategy was working very, very well, and we saw a very great reduction in the M2 frequency, and it accompanied an increase in the M1 population in the tumor-bearing animal, and which was also functionally validated by the positivity of iNOS, which is a classic M1 marker, and a decrease in arginase 1, which is a classic M2 marker. And then this translated very well to the CD8 and the CD4 activated cells. And we could see that this strategy is causing an activation of the CD8 T cells. And we see the tumor lysis very, very efficiently in the tumor-bearing animals. So we wanted to validate this in the human samples. So we initially started by screening doxycycline for the cell line, which are either TNBC, ERPR positive or HER2 positive. And later on we took the tumor biopsies, and we could see that the triple negative breast cancers have a higher abundance of these ectoenzymes on their surface, as indicated by the green and red fluorescence. Similarly, the infiltrated macrophages, which is 11p positive macrophages, also had higher expression of these ectoenzymes. So we then took the peripheral blood from these patients which were either TNBC or HER2 positive or ERPR positive, and then drive the PBMCs to macrophages and treated them with doxycycline. And as we can see, doxycycline was able to cause a decrease in these ectoenzyme even from the PBMC-derived macrophages. And similarly, we could translate the same even into the T cell, which are derived from the patient's tumor-bearing patients of different categories. And when we combine doxycycline with the antagonist, particularly the antagonist of CD39, which was much more potent in the preclinical model, we could see an immunoactivation and a lot of synergy in TNBC as well as HER2 positive patient. We could not do much about the ERPR positive because the results were not too encouraging, hence I'm not presenting them here. And finally, we could really zero down to the fact that in the TNBC patient, the effect of doxycycline and the antagonist is much more pronounced in the TNBC patient compared to the HER2 positive patient, presenting doxycycline as a very, very novel immune checkpoint inhibitor. But in this case, we had to combine it with the known inhibitors. So now we also did it for CD4 activation. Now what we plan to do is we plan to develop certain analogs of doxycycline so that we won't have to utilize the antagonist, and doxycycline by itself can be a monoimmunotherapy for TNBC patients, or we would go for developing a formulation for sustained release, because doxycycline is highly hydrophilic in nature. So in order for a molecule to be acting as an anti-cancer, it has to stay in the system for a longer period of time. So we want to develop a formulation, and then we are also exploring whether doxycycline can also act as an immune checkpoint blocker for PD-1, PD-L1. So in summary, we really could provide a very early indication that doxycycline can act as a novel immune checkpoint molecule. So I would like to acknowledge GSBTM, ICMR for funding support, Dr. Abhijit Dey for the 41 cells, and my clinical and the animal collaborators from Nirma University. And thank you so much.
Thank you so much, ma'am. I would like to request Ms. Pankti Mistry to come up on stage to present a memento to ma'am. Please give a big hand, ladies and gentlemen. Thank you. Thank you so much. Now may I invite upon the podium Dr. Meera Sharma, Registered Patent Agent and Scientific Advisor with the Indian Patent Office, to address the audience on technology and IP scenario of state. Dr. Meera Sharma is a Registered Patent Agent and Scientific Advisor with the Indian Patent Office, specializing in patents, designs, and intellectual property consulting. With a PhD in immunopathology and advanced diplomas in intellectual property law, she has over 2 decades of professional experience in IP strategy, patent facilitation, and capacity building for innovators and institutions. With a huge round of applause, ladies and gentlemen, please welcome Dr. Meera Sharma on stage.
Thank you, Manali, for a wonderful introduction. First of all, I would like to thank Dr. Anusuya Badalkar and her team for organizing such a wonderful conclave. And since morning, we are all witnessing insights into pathogenesis, prevention, treatment, prognosis of the cancer. My topic is a little bit different. It is based on the technology and how to materialize on the academic achievements of all the academicians who are present out here. So I have been given 5 minutes deadline, but I'll take few minutes extra. Please forgive me. How to use it? Which one?
This for forward. And this for back.
Okay, so now I I will skip many of my slides, which will unnecessarily take up more of your time. But this slide I would really like to highlight upon, because as academicians, we always focus on publishing our papers. And we are actually disclosing the ideas which many people materialize on patenting it and commercializing it. So I'm just extrapolating the idea of Dr. Purnima, Dr. Paini, and then Dr. Muthurasu, that we should go ahead with technology, legal certification of the technology and saving the technology which belongs to us, which we have devised. So the patents, first file a patent and then you can go ahead with publishing your papers on the findings of your patented technology. Or even if you have applied for patent, even if it has not been granted, please go ahead and publish the paper thereafter. Don't wait for the patent to be granted to publish your paper. So that is my professional suggestion and advice to all the academicians who present here. So now next is IPR. I will skip this slide. Everyone knows as of now what IPR is all about. It's all about patents, designs, trademarks, copyrights, etc. Now this is a thought process which goes on in the mind of every scientist, that what is the problem we are solving and whether it is a product we are devising or a process we are devising. So we have both types of patents for the products also and for the process also. And we just have to take care of three things: novelty, inventive step, and the industrial application, which is very important. Otherwise, your technology is not going to be granted patent unless and until it is industrially applicable. We should be able to commercialize it. And that is what inventive step means. Involves technical advancement as compared to the existing knowledge, having economic significance, or both. And now these are the basic principles which I will not really go ahead with. Of course, this is for both the product and the process patent. We can commercialize by assigning the patent or by licensing it, or the patentee himself can scale up the product or the process and sell it in the market. It should be sellable. It should be vendable. So this is the problem solution approach. See, this is the conventional bicycle parking, which is horizontal. It's taking up a lot of space. Now, if we want to save the space, we can have a vertical parking. So this is, in a simplified way, I have told you what the solution should be to the existing problem of space. Likewise, for scientists out here, I have seen a lot of problems being addressed, but they are mostly academic. So I would request all the scientists, please focus more on the technical aspects which can be materialized, which can be commercialized. Now, this is important because this information, since morning we have had the statistics of the occurrence of the cancer, different types of tumors in India, in world per se, in Gujarat. But this is the patent data. How much patent filings have been done in India? And Gujarat is, you can see over here, it is 8th in number. See, here is the Gujarat. So we need to push up our patent filing trend. We are doing immensely good work. I've seen the almost all the presentations today. We are very good scientists as it is. But we need to file more patents, get them granted, and as one of the speakers said, I am a Gujarati, so I would think about making money. Please think. All of you, think like Gujaratis when in Gujarat. So, next is the patent grants dashboard. Again, the same scenario. And now from this patents grant and patents filing, it is not just limited to cancer research. It is overall view. And these slides, this data I have got from Indian Patent Office website. It is not my generated, it is their copyrighted matter. Okay? So now coming to the cancer research is not a new thing. It is it originated in 1893 and still going on. Still we are struggling to find solutions to prevent, to treat, and to have a better prognosis in the cancer patients. So, see, a lot more has to be done. Of course, the patent filing in the cancer has started from 1962 when the interleukins were seen as one of the promising candidates to curb the cancer progression. Okay? So, now the major technology areas of cancer patients, most of these have been dealt with, like anti-cancer drugs and formulations, diagnostics and biomarkers, medical devices and radiotherapy, biotechnology, molecular and oncology. But the latest trending one is the digital and AI-based innovations. And I'm glad that Pahini today gave a very fairly good view of the AI-based technology, how it can be used to diagnose cancer at an early stage. And Dr. Muthurasu also dealt with some of the bioinformatics data in GenX AI. And I'm glad that I heard your presentation. I was very much impressed. And next, of course, major patents relate to incremental innovations, formulation process patents, and device-related patents. And it gives me very pride feeling to tell you that in March, I got 4 patents which were granted to AI-based medical devices. But that was for diabetes and infectious diseases, none for cancer detection. So please, kindly, my request to all the scientists out here and to Madam Director, Dr. Anusuya, please insist the inventors to focus on AI-based innovations in cancer research. This is my genuine suggestion and request to you, because that is where the future holds. We are lacking far behind the Western world in that scenario. That you will know just now. Now, the technologies which have been developed in Gujarat, that was my main topic which I was to stress upon. Of course, there are many technologies which have been patented in Gujarat. It's not just that we are lagging behind. We are 8th in country in getting the patents granted. So that's fairly good enough number. But what about in oncology? In oncology, I have listed only these many patents which have been granted. Of course, some of them have been funded by GSBTM and MS University, then Gujarat University also. ITM SLS Baroda University is doing very good job. If you see their website, they have immense number of patents, especially mechanical engineering, electrical engineering, and oncology. So that is, if anyone from their institute is here, I must appreciate it. And then, of course, GBRC is doing good. It is having 2 patents. Then Ahmedabad-based this Cadila is having 1. So these are the patents specific to cancer diagnosis. And the something which really impressed me of ITM SLS Baroda University was, this was the one patent which was using AI system for breast cancer detection, which was not seen in any of the patents which were granted in Gujarat. I'm not talking of India per se, because my topic was limited to Gujarat in the state I had to focus on. So this is one, we should take inspiration from such kind of patents. The number is also there. And if anyone wants more details, you can always get in touch with me. I will give you as many details and support as much as possible. And now coming to AI in cancer care, this has already been discussed by Dr. Pahini in the morning. So enhanced diagnosis, personalized treatment, then clinical detection support, biomarker discovery, drug development and designing. I think Dr. Muthurasu also told how the bioinformatics data and the sequence analysis can help you directly, instead of wasting months in getting sequence analysis done. Please get to the bioinformatics professional and get the data. Dr. Purnima is here. Thank you for coming over, ma'am. I'm insisting on the patent importance in our research work. So these are the various gray areas where we should be working on in cancer research. That is AI facilitated imaging diagnostics. This I was mentioning some of the scientists in the morning about NVIDIA. that please go through the website of NVIDIA. There they are using quantum mechanics to diagnose the cancer at -1 stage, when it is not even showing any kind of cellular expression change. Also at the mitochondrial level, a single protein conformational change is diagnosed by quantum mechanics, and that is all AI-based MRI system which they have developed. Please go through the website of NVIDIA. So this is where the AI technology is taking us to. In super early detection of cancer, I should say. It's -1, not even the cellular markers are expressing. So then is genomics and the biomarker discovery, which Dr. Muthurasu told, and then AI facilitated liquid biopsies, then 3D visualization, radiotherapy, robotic surgery, therapeutic inventions. All these are there. But there are certain, of course, these are the various steps where the patient cancer journey can be affected by AI-based technologies. At every step you can see over here, when a person is asymptomatic, then also you can detect. Just go for a routine MRI scan, which is using AI-based systems to detect even a single molecular change, intracellular change. Then we can go to the disease detection, diagnosis and classification, then disease and organs at risk segmentation, then others like imaging optimization, automated reports, then complex data integration, then also assisting in the robotic surgery is a very common thing now. There are sustainable development goals in personalized cancer care, then issues and concerns in using AI. This is very important. Why AI is not as of now very, very popular? Because of these limitations, that is difficulty of standardizing, gathering and managing data, the big bias present in training sets, the absence of strong reporting guidelines, the relative dearth of prospective clinical validation studies. I think Dr. Muthurasu will need to give attention to this. Difficulties implementing user designs and workflow, antiquated legal and regulatory frameworks surrounding AI, and the exponential growth of knowledge and dynamic data. Where all and what to look. If you go for sequence analysis, you have n number of sequences. It's endless. The list is endless. The sequences are endless. So which sequence to target to? So how to shortlist that? So there AI-based data will help us. Now coming to digital healthcare, this is an example of bionic lens. And this is a very recent concept for India. I don't think anyone has ever developed theranostics, madam, therapeutics and diagnostics together. This is another gray area where we should float some funding and trigger these scientists' intellect acumen. We should go for theranostics. So we should float grants for theranostics development, and it is already there in diabetes. There is a small patch which is put on your wrist, which simultaneously detects the glucose level of in your blood, and likewise it also injects insulin into your bloodstream to keep your glucose level normal. And if it is above 120 or 150, whatever the range has been set in that particular patch which has been put on the wrist, it will control your blood sugar accordingly. So that is a theranostic. It is side by side detecting your blood sugar and also giving insulin side by side. So this concept people are using in India, but those things they are getting from US or Russia. Now coming to the fabrication of theranostic devices, that is beyond the scope of this conclave. For that I think semiconductor mission will be of better use in this. And yes, biomedical scientists, my fellow colleagues, please interact with computer engineers, because it is interdisciplinary science now. It is not limited to biotech science or any life science. Now I am getting a red flag. So these are immunotherapeutics in cancer. So I'm simply going through, there are 20,611 patents as of now, but what about patents in Gujarat? big question. Please focus on that too. So these are the current owners of the immunotherapeutics. These, you can see, are mostly the companies abroad, and they are getting it patented in India. So they are earning from us. Then recent advances in challenges, this has already been dealt with earlier. And then CAR T cells therapy. See the companies which have got the patent. None in Gujarat, only one in India is there, but it is still not been granted. It's still in the pipeline. Then CAR T cells therapy of cancer, current legal status is that this is the international status. This is out of bound, but from my topic, it was to focus only on Gujarat, but I didn't have the data for Gujarat. Just to motivate you all, please let us live up to these standards which are being set by other people in other countries. So this is immunocheckpoint inhibitors, and this is the pie chart which shows the distribution of patent filings based on the types of assignees. You can see corporates have the major chunk of it instead of the individual scientists. So these are the top 5 healthcare startups in India which are actually working very well, and but none from Gujarat, most of them from Bangalore. So let us please gear up our arms together and our brains together to put Gujarat on #1 position in India. Thank you very much.
Thank you so much. I'll request Dr. Nitish Dave from GSBTM to come up on stage and present a memento to ma'am. Please give her a big hand, ladies and gentlemen. Thank you. Thank you so much. With the technologies being developed and transferred and commercialized, where is it that we would want the technologies to reach? How do we meet the unmet needs of the society? What more needs to be done for the epidemic of cancer? To discuss these problems and the challenges and the problems' probable solutions, may I invite upon the stage our panelists for the panel discussion on possibilities of translational opportunities for funded projects. This session brings together esteemed representatives from BCIL, Fridge Institute of Human Genetics, Tata Memorial Hospital and Kerry Hospital to explore how research can effectively transition from the lab to real-world application. We have with us, keep clapping ladies and gentlemen, Dr. Harsh Sheth, Fridge Institute of Human Genetics, Dr. Purnima Sharma, BCIL, Dr. Rohan Chaubal, Tata Memorial Hospital, keep clapping everybody, and Dr. Somesh Chandra, renowned oncosurgeon at Kerry Hospital and principal advisor at IOTA Diagnostic Private Limited. They are joining us as the panelists, and guiding this insightful conversation is our session moderator, Professor Dr. Rakesh Rawal, Dean and Professor at Gujarat Biotechnology University and a renowned cancer biologist with over 3 decades of research experience. May I request the moderator and all the esteemed panelists to kindly join us on the dais. And by the time they are getting seated, I'll request all the speakers to be seated in the front row, in the front sofas, so that we can easily, connect with them.
Good evening. Though there is a thin crowd left, but we require a sensible crowd. So thank you all for being with us. So the whole conclave was conceptualized because whenever we go to the principal secretary ma'am or minister, sir, so they ask, in spite of so much funding, why we are not coming up anything in this field, specifically cancer. And after looking all these, you all agree that we have good researchers individually, but somehow the dots were not connected. So the first objective of this conclave was to connect those dots. There is a basic scientist, there is a clinician, there is a pathologist, and there is a synthetic biologist or there is a pharmacologist, but we are not talking to each other at the same platform. Individually we will do great, but in silos. And possibly that's the reason that we are not meeting or matching our national output in terms of patent or IP or anything. And second thing, we researchers, we believe in publishing more than patenting. And we represent a country of around 146 crore with cultural diversity, with genetic diversity, and with different access to healthcare facility across the India. And therefore, we have to invest much on, so if you look today's presentations, we started with prevalence, we have early diagnosis, we have late interventions, we have advanced therapeutic modalities, including CAR-T and all these. So we covered the whole spectrum which is there. So the first intention is to collaborate and connect academician with industry as well as with clinician. So until these three will come together, we will not be able to produce something which is impactful. So with this, we received several queries from participants, and there were queries from the government also to discuss few things. So the first query that comes to my mind is, in India, how cancer can be mapped across the population and which technologies can be useful. So I request Dr. Somesh to focus something on this.
So basically, if we go by the conventional classification of cancer, that's how it is being mapped till now. Because it had a lot of epidemiological and importance in framing policies. But now oncology is becoming tumor agnostic. And if we really want to map things in a futuristic way, we need to start setting up those kinds of maps, molecular maps, how practical this would be. And I mean, we need to brainstorm on this. I guess if you look at TNM staging, it is basically an anatomic staging, anatomic classification of cancer. But over the years, it has progressively incorporated biological markers like grades and tumor markers and such things. So there's a way to preserve the basic identity, the basic structure, the basic DNA of cancer classification, and then maybe add these features to it. I wrote a blog recently I write blogs for Indian Journal of Surgical Oncology, that we have lost our tails, but we have not lost the tailbone. So how to preserve what needs to be carried forward and how to add on to it, that's something.
So at GCRI also we initially started working on or looking at the patient cases. Where do they come from and what type of job they were doing. So if you try to do a cross mapping, we will come to know possibly what are the etiological factors, what are the regional distribution of these cases across the population. So once we know that, possibly we know the etiopathogenesis of the disease and we can think of some intervention. So a hospital in Rajasthan receives thousands of cases from Punjab, and when you look back, possibly you will go to the root that was because of certain pesticides or insecticides they used in farming. So such mapping is required across India, that which area is prone to which type of cancer, and then possibly we can devise some preventive measures for that. So being a clinician, how you think that where India stands in adopting precision therapy and targeted therapy at present?
So far as advising such kind of therapy is concerned, it's very easy because it's all available on your fingertips or on your thumb, just a click of thumb. You have, patients are now, I have a patient admitted who is continuously on any kind of NLP, and the thing is, what we call personalized precision medicine is largely at present drug driven, and everything is not available. Everything is not proven for each kind of patients. The Memorial Sloan Kettering some time back, they published, and there are other such registries, something called OncoKB, that's Onco Knowledge Base. So that relates to what kind of molecular targets can be pointed at in different kinds of clinical scenarios. Because like someone said in the morning, we just find a mutation or any genetic targetable agent, just we go after that. But then there are other associated changes, germline changes. So yes, we are in a different sectors have got different adoption rates. The rich and the privileged, I think they have all the kind of access to all kinds of precision medicine. But by and large, it remains inaccessible to large masses.
In continuation to that, I would ask Dr. Seth to throw some light on the recent advances in molecular pathology or in support of precision medicine and diagnostic. How equipped we are currently in India, or we can say in Gujarat, for this?
So I think, sir, very nicely summarized. We've gone from the era of looking down the microscope to slowly doing cytogenetics work to now doing sequencing. So there are certain tumors where you wouldn't even begin the treatment until you have identified a genetic marker so that you can give a targeted therapy. And very recently, CPIC guidelines and DWPG guidelines also came up regarding 5-fluorouracil, because you can sometimes have adverse reaction if you were to give 5-fluorouracil. So all of these guidelines are now coming up. The adoptions are happening as well. I don't think sequencing technology in Gujarat is lagging behind in any way, shape or form. Whether you go to a government hospital like GCRI or civil hospital, which can be supported by private industry or by government itself or private entities by themselves. I think sequencing is not that difficult anymore. It is the interpretation where the challenge comes in. And I think Sir really nicely summarized those challenges. The biggest problem you would find, in my humble opinion out here, and I'm sticking my neck out, is when you have a 50-page report in front of you. If you were to do a 2 lakh rupee, 3 lakh rupee test, comprehensive genomic profile, and Rohan is smiling right next to me, because it's a constant discussion we have with clinicians. How do you interpret a 50-page report? I simply want to know what drug to give. I don't want to write a thesis on it. So the next challenge is not sequencing, it's interpreting what is right there in front of you. Whether it is the context, a broad context that Dr. Pandey really nicely mentioned, that you don't look at just one part but see what's happening downstream, or you understand the basic biology and know which drug works very well against that basic biology. So MSI is a wonderful mark of that. So this is where the challenge will lie. And it's not just challenge of Gujarat or India, it's a global challenge. And it has to be resolved, and this is where the frontiers will start emerging in front of us.
So we need to focus more on clinically actionable defects rather than
yes, sir.
The complex reports.
Yes, sir.
So being the translational conclave, I would request Dr. Purnima to throw some light on what are the barriers of translating research into markets.
So the main barriers is the disconnect with of the academicians with the industry. So actually, that disconnect has to be bridged. That is number one. The other barrier that I immediately can think of is the funding. So the funds that flow in are for discovery. They are just limited to discovery. Whereas when we look at cancer, it has to extend even to deployment. So at least whatever is done, it should extend for validation. Like they say, lot of data has to be generated, and all that data will have to be integrated, even if we have to look at AI models, etc. So I think the funding should not just stop at discovery, but it should also extend to validation, so that the scientists are in a way forced to, go to clinicians and for deployment of, or at least for validation of whatever has been done, so that the applicability of all the discovery is then, gets taken care. And then probably the scale up, and from there onwards, I think the TTOs can come in. They can help in, further putting all that data, the validation and the regulatory and the market, etc., so that they can at least make the industry to appreciate what is done, and enthuse interest among the industries to come in. Of course, in many cases, we've seen that even the regulatory guidelines are not there. So I think bringing in the policy makers to look at those. And since morning, we've seen in some of these new technologies, modern technology, even the guidelines are not there. So that is, of course, there's something which has to happen parallelly. But these are the, broad, I would say, disconnects which needs to be bridged. So I think, to cut the long story short, it is the gap between the academicians and the industry which has to be bridged. And that also involves, I think, scaling up the government, policy makers, market, and also even the deployment, because sometimes it has to be deployed at the laboratory level, at the public health center, at the hospital. So I think all that will come in, but that is a little far-fetched. So gaps are there, but immediate gap, I think I would say the funding should not stop at only discovery. It should also go further little for deployment.
True. So now coming to the funding, I would ask Rohan to throw some light on at what level of TLL usually either the venture capitalist or all those industry will be interested. Because if you take the life cycle, ideation to prototype, and then you need validation. So at what level you think it should reach? Raise once.
Hello.
Yeah.
So I would suggest it's a misnomer right now that there is no capital flow in the market. I think that's one of the biggest misnomers that we have, and we need to get past that. The liquidity infusion in India's capital market is second to none. And this liquidity infusion is across all stages. So if you look at our funding that comes from the state, it is perhaps one of the most well characterized and designed programs. You have funding to get you from the ideation stage to the pre-seed stage. You have the MeitY EIR grants that supports a founder even when they have an idea and they don't have a product. I think India is one of the few countries in the world to have that. Then you have funding for the pre-seed, you have funding for the seed. The problem with this is the way the funding has been disbursed. You get funding announced in year 0, and then the money actually comes to your bank in, let's say, year 1, if you are lucky, in year 1.5 or second year. The bureaucratic process to get the money into the account is what leads to failure for most startups. And in this regard, I think we have to give it off to GSBTM. They are very, very diligent with their funding, the projects that they have supported. And in fact, we have her sitting here. He is one of the few people to have actually got a funded project and a laboratory developed test to market completely with government funding and support. And that is a shining example of how you can execute it. So to answer your question, venture capitalists should come in only when you're looking at scaling up or when you want to expand outside India. If you are looking to just take a product from, let's say, TRL 0 to TRL 4, you have adequate government funding, find the right partners. There are lots of incubators, lots of funding agencies. If you are wanting to go from TRL4 to TRL6, again, lots of funding available in the form of conducting clinical trials. TRL6 to 9, wherein you need to hire staff and where you have shown that the MVP exists, look, this works, the clinical validation has been done. That is where typically a VC should come in, because that is when you need to hire your marketing team, your sales team. The problem that most of us have, and I would blame a very popular reality show that has converted the art of entrepreneurship to something like valuations game. What that show has ingrained in us, that we need to go out and we need to ask for funding just to build an MVP or a prototype, which is not the case. Venture capital by its very nature exists to scale things up and to bring in capital when you are scaling from 1 to 10, not 0 to 1. We have adequate infrastructure in the country, adequate funding to go from 0 to 1. It is just that our bureaucratic processes have to be improved.
So true. So we are fortunate that we are in Gujarat and we are being supported by GSBTM from ideation to any TRL level, and ultimately even there is support for patentization if it is from the project. But there are certain layers of barriers which are there. One initial barrier is the bridge between academia and clinic. Second is between academia and industry. So how to break those barriers? So you can take help of any participant.
I will briefly, take this up, and I could go on all night. Harsh is very much aware of where I come from, but I will not bore the rest of you. But we have to understand the distinction between India and Bharat, and that is very, very true. I am based at a hospital in India that looks at something like roughly 27 to 29% of all cancer patients in India. If you look at the patients who come to us, they come from all different economic strata, all walks of life. But what is striking is that everyone who comes to us, most of them come at a very advanced stage of disease. They present at stage 3 or stage 4 when the disease has progressed. What that means is that, because we do not have very robust screening programs, most of our cancers are at an advanced stage. The products that we are developing today are not what Bharat needs. They are maybe what India needs and maybe what the West needs. So when you talk about an AI for screening or early detection of cancer, or let's say genomic tools, let's say a liquid biopsy, the problem is most of our cancers are already at a fairly advanced stage wherein they are most likely resistant to therapy. The tumors are very heterogeneous. You have multiple hits, so single drug entity may not work. Those are the sort of challenges that we need to address. Unfortunately, that is not happening, and this is not an industry academia problem. This is a problem with understanding what are the real needs of the country. This, I would probably say, stems from not talking to clinicians and not going to hospitals. This is something that has to change, and I believe the first step is, we should start with our undergrad and master students who in this field need to go to a clinic, spend some time in a clinic, work with clinicians, understand patient flows, understand how a clinician decides what sort of treatment is needed. Because unless you sit there, you don't understand what our problems are.
He's spoken my words that if someone is, when I'm sitting in my clinic, there are questions in my mind arising continuously. And those are the questions which actually matter to the patient, the answers to which matter to the patient. And I have no answers to them. So it's the academia which can find solutions and the industry which can bring out, show them the light of the day. If you look at industry, primarily there are two. One are medical equipment manufacturers and the pharma. I don't know how much of a clinician, at what stage a clinician in India is involved in these industries. Currently, in my understanding in India, a clinician is a passive end user or whatever is provided to him or her. And that is why we are not making radical changes here in India. We are just borrowing it from outside.
So, yeah. Other than that, madam, you can add the regulatory and technology barrier also, other than these two barriers.
Yeah, I would also, just to add to that, I fully agree to what has been said by other experts. In case of cancer especially, even at the idea generation stage also, I think it is important that the scientists, the clinicians and the industry should, work together to understand the problem and also to design the whole, research so that it is focused with the deployment in mind. So that is very much required. Otherwise, all these three segments, they'll keep on working in silos and nothing comes out, which is what is the case, which is happening now. Now, as far as the regulators are concerned, it is difficult, to basically just like that go, because unless we are clear of what is the product or the technology, we have to see whether we have the regulatory guidelines there, and if they are there, then how to go about. Because if they are there, then we just need to have those protocols, how to generate the data as per the protocols, where those data has to be generated. Because I've seen presently also that there are, whenever the clinicians are also collaborating with the scientists, the data which is generated it is basically to satisfy the scientific interest. It is not from the perspective of addressing the regulatory requirements. So that's why, again, if we have all these people coming together and also the TTOs, because regulators would not come on board right from the beginning to design the research, we can help to ensure that whatever data is generated, it is from the, it is in compliance with the regulatory requirements, and then ultimately it expedites the technology deployment.
Thank you, ma'am. I have to relieve you because you have to fly to catch. You have a flight to catch. So we'll continue that. So possibly that was one of the intention why this conclave was made, because we thought of listing out all clinicians who have got inclination or contribute towards cancer research. So possibly at the end of this, we will have a list of hospitals or clinicians who are inclined to work along with the basic scientists or industry, so that once we have all those dots connected, possibly we can bring something which is meaningful and impactful. Yes, you wanted to say something. Sure.
Thank you, panel discussants, for this bringing up this very important aspect of funding and the diagnosis and the clinical treatment of the end result of the cancer research. Now, for funding, my one of the points which I left during my presentation was, in my last 18 years of as a IPR consultant, I have got 116 patents granted. And out of those 116 patents which have been granted, I have got 57 of them commercialized. So ROI comes within few months' time to the academicians which they have spent on research and development and getting the patent, covering the patent cost also. So apart from the government institutes like schemes like MSME or GSBTM or other funding schemes, this is another thing which we can look into, not just by sitting on the patents if they are granted. Okay, we are just illuminating our CVs that okay, we have so many patents granted to us, but how many of them have actually seen the light of the day? How many industry has picked up to cover a solution to a problem? That is my one suggestion, that patents should be commercialized, which is a very big gap, and that is what I am also trying to do. It's a hard work, but in the end it's very rewarding. Another thing about the biomarkers, sir, which you said, recently I have got one patent granted for Actrac, which is based on stem cell therapy. And when we go for umbilical cord MSC conditioned media, which has been supplemented by various growth factors and interleukins, it does not target any biomarker. It immediately targets various tumor cells which are growing. And it has been shown in vitro and in vivo also, in the clinical trials are also going on. It is now into, I think, phase 2 clinical trial in ACTRAC. So that is one more thing which I understood, that biomarker is not the only thing which we can focus on. We can always shift our focus to stem cell therapy, which is showing very promising results in tumor therapy also, and in hair regeneration and cosmetic applications too.
Thank you.
Thank you very much.
So if I may just answer that, you see, sometimes we have to get past this what I would call a very Indian obsession with patents. What we have to understand is innovation does not equal to needs. So very good example. You have two people sitting on the dais right now who have repurposed therapies, not patented. Harsh has worked on one of the most pivotal trials in the past decade on the aspirin trial, which has actually resolved this whole conundrum that aspirin brought on the scene. He has shown unequivocally aspirin helps, not patented. I have worked with my mentors on two of the most pivotal trials in the past decade in breast cancer, wherein we have shown that a single shot of hydroxyprogesterone, which is 100 rupees injection given prior to surgery, improves outcomes by up to 12% in node-positive breast cancer patients, and which is now the standard of care at Tata Memorial Hospital and all hospitals on the national cancer grid. A 10% or to 12% incremental improvement in node-positive breast cancer disease is one of the largest improvements that has been shown. Not just that, just 2 years ago we showed that a single shot of lidocaine injection, which blocks VGSC channels, voltage gated sodium channels, leads to an improvement of 14%. If you add up, so there is a clinical trial that is now ongoing for progesterone plus lidocaine, and we are trying to see the additive effect. Again, not patentable. So where I'm coming from is, yes, having patents is nice, having innovations is nice, but a country like India which needs solutions tomorrow, there's a lot more that can be done with existing innovations, drug repurposing. It's just, our creativity and imagination that needs to work. I mean, that can also, these are low-hanging fruits that can lead to great improvement in outcomes, and the government and funding agencies also need to support this sort of creativity and innovations. So one of the things that funding agencies always ask, or venture capitalists also ask, what is your moat, what is novel. I mean, these sort of things do not have moat, but they lead to a larger social good which should also be supported.
So at GSBT we don't ask that stringently, but yeah, definitely to, I think, go ahead successfully. If you look back, why we don't have handful of good data which can be translated. So the suggestion that came from advisory body and technical experts that we should have a Gujarat Cancer Genome Atlas and be part of Indian Cancer Genome Atlas. But for that, what we need to do, we need to have a good tumor repository with good biospecimens, and whatever projects that comes out of that, if they share the same similar resources which are clinically annotated, and if the data comes, it will have some meaning and it will add some meaning to that. So do you agree to that?
So, from a geneticist standpoint, because we are talking about somatic expansion, all the data that we have seen so far, even today, taking example of Lynch, there's no difference in genetic architecture. If we take autism, right, there's no difference in genetic architecture. If we take male infertility, the paper is going to be published next month. There's no difference in genetic architecture. We can have as many atlases as we like. It's nice to have a good data repository, but the evidence that we have so far show it, when it comes to somatic or diseases where de novo variants are primarily the cause, the genetic architecture is not that different. So having major biobank repositories and atlases actually don't help that much in a grand scheme of things. They would help when we are doing a population scale genome project, right? So your 100,000 Genomes project in the UK or All of Us program, but they're more for reclassification of VUSs. For the somatic variants, the jury is
But we got
not
good
that
results
straightforward.
in germline changes for breast cancer. I think we had clear-cut evidences that somatic will show something, but germline have still some lineage to show the susceptibility.
So just to add to what Harsh is saying, see, I've had the opportunity to collaborate and contribute to two of the largest ongoing genome studies. One is the Indian Cancer Genome Atlas, which is a private consortium, and the Indian Breast Cancer Genome Atlas, which is a CSR effort. When you look at the data that comes from both the projects, I mean, there is a very, very small, minute tail of somatic mutations that are maybe different from, between the Caucasians and us. The predominantly overwhelming 80 to 90% of the variants are the same. Now, the question this begs is, for this small 10% of delta that you see in a very small subset of population, is such a large expenditure justifiable? Where I would, second what Harsha said is, instead what we need is, given the way we have consanguineous marriages, the way our population structure is driven by founder mutations, it would be much more pertinent to look at the germline space and identify what are the drivers of genetic or hereditary diseases. India has perhaps one of the largest undiagnosed populations of hereditary and genetic diseases, and no one is talking about it. You see, we are so enamored with cancer. I mean, there is absolutely nothing different. See, at the end of the day, we are not aliens. Somebody who gets breast cancers in Connecticut will have the same etiology as maybe somebody who gets in Mumbai. There may be minor differences. But when you look at somebody who has some other germline hereditary diseases, this may be something that are peculiar to India. And these are not things that the West is going to look at. These are our people, these are our problems. We have to solve for them. So I think somewhere down the line, our focus has to shift from genomic to germline and genetic. And I think GSBTM has been doing an excellent job looking at that.
Yeah, so germline is part of this. So I think with this, we need to conclude as we are running out of time. So any quick remark if you want to make 1-2 line each, that's fine.
This obviously not my domain. Like I said, I work at the end of the line. But there are a lot of questions which come to my mind when we talk about, say, gallbladder cancer. We have still not been able to really understand why it is so common. It's like an epidemic in that Indo-Gangetic belt. So maybe a genome-wide study, GWAS might show us something which is going on there. We need to possibly integrate different kinds of data. So enterobiomes, we have not probably been looking quite enough of how they interact with the germline or the genomic changes. So if we have this kind of a multimodal data map, probably we can have some more meaningful. Problem with clinical data is that it's very inaccurate at present. It's very incomplete. And building a model on the current system is really hazardous to my mind. What we first need to put in place is a very good EHR kind of a system which can be shared across all hospitals. Some good institutions have their own, but it has to be so much objectified that the chances of error are very little. At present, what is being captured in most places is in a PDF form, and we need to convert that into a more synoptic form so that data capturing is uniform, data analysis is easier, and then integrating with other genomic and multi-omics is better.
2 very quick points. First one, we saw therapeutics is exciting. Diagnostics, really, really exciting. Prevention, not so sexy. Think about this trip, right? for assessing cervical cancer or HPV risk, or talking about aspirin or 2 slightly unripe bananas every day. I mean, who talks about these things? The thing is, they're exceptionally important because, number one, they help in driving the burden of cancer down. And number two, the average cost of cancer care, this is average cost, 5 years ago was 10 lakh rupee per patient. By you spending a small amount of money upfront to prevent cancer, you're making a long-term investment in your own health and the health of the nation. So prevention is front and center, and that as a geneticist should be ingrained. Number two, the interaction between scientists and clinicians. I know when I started my journey here, first 2 years were exceptionally challenging because you are sitting in a clinical setting. There are things that you're not aware of and you don't know. Don't be shy. You'll be slapped around on your wrist a couple of times. But after some point, you will start to understand the lingo. You will have a shared language. And trust me, it will guide all the fundamental research you will ever do in your life. So please spend those 1 or 2 years in a clinical setting. It will change your life.
Yeah, my 2 cents are, we should stop, for lack of a better word, innovation shaming. Indian pharmaceutical industry is one of the greatest assets that India has. Our generics industry, people who have not taken healthcare outside India don't know how good they have it in India. The drug prices, the easy accessibility, affordability of drugs, that is our core strength and we need to leverage it. Example that I would give for this is Keytruda, pembrolizumab, the largest selling, most profitable drug for the past decade. Keynote 63, which came out in 2021, established immunotherapy as the standard of care across a couple of cancers, and then it flowed through that. What you have to understand is, in cancer, drug development takes many years because of the way the endpoints are structured. So we very frequently have 3-year follow-ups, we have 5-year overall survival, and in some cases we even have 10-year overall survival. So once a drug comes out on the market, till the time it becomes an indication, very often the patent ends up expiring. This is what has happened with pembrolizumab. Now it is in the prime of its life. Many of the clinical trials, which are very, very expensive trials, have established it as the standard of care, and the patent expires in 2028. Now think about what would have happened if we would have gone ahead with an innovator drug trying to do this. We would have wasted these billions of dollars in trying to prove that this drug works, conducting expensive clinical trials. Instead, now what we have is a blockbuster drug handed out to us on a platter, for which the Indian generics industry is poised to, make biosimilars to copy Keytruda, and the next decade of onco care is going to change because Keytruda is going to be easily available, it is going to be affordable, it is going to replace chemotherapy. So there is a reason why you see daycare chemo centers cropping up every now and then in the news. Why oncology is so hot? Because immunotherapy is going to change the way cancer care is being taken. So sometimes it is good not to be an innovator. We should leverage our strengths and we should look to be creative, innovative, look at repurposing what we already have instead of just keep on crying that there is no innovation. I think that's an important thing.
True. So do the best in the available resources you have, and try solving our own regional, national issues and challenges. But for that, we need to talk to each other. Researcher has to talk to clinicians to get their problem statements and try to answer that. And ultimately, we need to collaborate. And for all this, wish you all the best. And thank you for being here. Thank you, all the panelists.
Thank you. Please give a huge round of applause. I'll request the panelists to stay up on stage for a few minutes. So it's now time to felicitate those who are yet to be felicitated. So I'll request Dr. Ansuya Bhadalkar, Joint Director R&D GSBTM, to come up on stage and present a memento to our moderator, Dr. Rakesh Raval, and our panelist, Dr. Rohan Chaubal from Tata Memorial Hospital. Zordar taliyan baja dijiye. Come on. Thank you so much, sir, for a very insightful panel discussion. Thank you. I'll request Dr. Rohan Chaubal now, please. Thank you. Thank you so much. And of course, I'll now request Dr. Ansuya Bhadalkar to deliver the closing remarks and formally conclude the conclave.
This mic is working. This is working. Thank you.
Good evening, everyone. I'm audible, right? Clearly? So it has been a long day. We pulled the sessions much beyond the time limits that we had put in our program structure. So I'm very sure that everybody now wants a break. You want your tea. I'll not stand between both of them. Very quickly summarizing the whole day's activities. So I was looking at it. I heard all the technical inputs that were there. And I was looking at it more from the perspective of a person who makes policies, who makes schemes, who makes concepts that can be then, inhabited by the researchers and scientists of the state. And from that point of view, I realized that all that we heard today is only a tip of the iceberg. There is much more that needs to be done, and there are a lot of possibilities that are still left open. More than finding out all the problem areas that are there in cancer research, I could hear as to there were a lot of suggestions, indirect suggestions, especially during the panel discussion, that how to do research. रिसर्च करिए लेकिन कैसे करिए वो सबसे ज़्यादा ज़रूरी मुझे सुनाई दिया and that also helps me understand when research proposals reach our office how to look at them and how to bring them into more focused work. So some things that I felt that we as GSBTM need to do is facilitate data exchange. The amount of work that has happened in the state and nationally, now that we are networked with a large number of people, we need to find out a common platform where we can exchange this data and we can connect the right kind of people to each other so that they can do something meaningful with that data. Second thing that I see is that the form in which the data is getting generated by the researchers. So there was a very clear input from one of our speakers that I think Dr. Purnima talked about it, that when you are generating the data, it should not be from the perspective of publication or patenting or something. It has to be from the perspective of delivering a solution. I have myself faced this in last few months that in the last few months that I have been working with the R&D portfolio, I saw a large number of people who put up such innovative projects which can lead to a technology. But when they write about the sample size that they are going to take to do their research, that is where there is a big challenge. So the sample size is too small, or it is too non-diverse to get any scientifically relevant data. So the work is good, the technology is good, the thought is good, but the planning is missing. So I think that is something our panelists very clearly emphasized on as to when you are writing a research project. Please focus on the type of data that you are generating so that whatever you do, it becomes either it can get into a very good publication or it can go into a real life patent that can be used later. And there was a very clear cry from our IP attorney who, very clearly said, whatever you do, if you are in the applicative research field, please do something that is patentable. That is one. And secondly, in India, all of the researchers and I myself, we know that Indian Patent Office gives patents quite easily, right? So that does not say that we have done something great that can reach the society. It is us as researchers, we as funding bodies, we have to keep a tab on what kind of research we are doing, where we want to go, right? If we want to increase the bandwidth of our ACR, then it is a different ballgame, right? But if we want to help someone in our own society where we have educated ourselves, right, and our taxpayers' money goes into it, then our call, our route of doing our research has to be different. So I think patent kariye, acha patent kariye, and please ensure that you commercialize it. If you believe in your patent, then you will see that it will get translated. Right, today we have BCIL with us, so all of you, you can write to us and we can connect your projects to BCIL to do an evaluation of your proposal, whether it stands a chance of good patent. Even if you do a patent, does it stand a chance of getting translated? Right, so write to us and we are right there to help you. I think these were some of the most important pointers that we collected from today's discussion. And Dr. Pavan Gupta very effervescently during his talk mentioned that there are certain diseases where India is leading, and oral cancer is one of them. So we should be the people who should teach the West what we do with oral cancer, and it should not be the other way around. So I think we need to intensify our work in it. And we have a lot of good work that has been done. We need to consolidate it so that we can give a prescription to the West and to the world. What do we do with the oral cancer data? So that is something all the scientists who are sitting here who have been working in oral cancer, maybe you people can come together and do it. GSBTM can do few more conferences. We cannot do everything. Right, but you know each other, and by today's conclave, you would have connected with a lot of people. And even with Dr. Pavan Gupta, who is at the center doing something in oral cancer, please connect with him, cross-connect with each other so that you can make a relevant data interpretation. Gujarat Cancer Genomic Atlas is something I think even state government will think about it. As our panelists mentioned, Dr. Harsh, Dr. Rohan, they mentioned that maybe we can take a better league or do something better. So I would invite all your opinions on should Gujarat have a cancer tissue bank, for example, right? So if not an atlas, should we have a tissue bank? In what form should we have? Should we have a physical tissue bank, or we should have also a digital portfolio, a digital form of that tissue bank as well, apart from having a physical tissue bank? So these are some very relevant questions which have come up through the day's discussion. I will request all the researchers to write back to us. All of you have my email ID. If you don't have, it's there on our website. Please write back to me as to what you think, that if that needs to be done. BCIL MOA is again something I will want to reiterate with all the researchers that kindly use this opportunity. It is completely funded by GSBTM. Your technology evaluation done by BCL, payment done by GSBTM. So there cannot be a more win-win thing for any technology transfer that can be done. And of course, our research support scheme is there, state government scheme. And there are a lot of changes in the guidelines that we have brought in. We have tried to make it a little more expansive, a little more clear so that you can involve national institutes in your research work. So please visit our website and study the guideline in detail. That will help you cull out better programs for yourself. I, however, was reading through different other conference papers. And today, after listening to everyone, I feel that there are certain areas of cancer research that we as a state can still work on, and that is neonatal and pediatric oncology. There the work is very less. New drug delivery systems, nanotechnology, and cancer prevention. Research in cancer prevention is something that we are not doing. Right, so we are addressing the disease when it has occurred and it has gone to a full bloom stage. But if we can work in preventive areas, that will be very, very nice. So these are some of the summary points, the pointers that I could gather. There would be more you would have gathered. So I request all of you to think together and bring out a good strategy for the state. And of course, GSBTM is there to fund. So we'll keep on doing that. Thank you very much. And I request everyone to join us for the high tea. Thank you.
Thank you so much, everyone. With this, we come to the conclusion of Cancer Research and Innovation Conclave 2025. I am Manali Shah on behalf of organizing team. Sincerely thank each one of you, the dignitaries, the delegates, the partners, everyone who has given their precious time today. Thank you so much, everybody. Networking tea has been arranged outside. Please have it and have a nice day. Thank you so much.